మేడం వినిపిస్తుందా మేడం? శ్రీదేవి మేడం.
అమ్మా నన్ను ఓకే
శ్రీదేవి మేడం గుడ్ మార్నింగ్ మేడం.
గుడ్ మార్నింగ్ రమ్మ, గుడ్ మార్నింగ్. పూజ, షెరిన్ మేడం కూడా జాయిన్ అయ్యారు. మేడం ను కూడా అంట కొంచెం కోహోస్ట్ చేయరంటమ్మా, అన్మ్యూట్ చేయలేకపోతున్నారంట.
Good morning, ma'am.
Good morning, Sharon.
Ma'am, is my screen visible?
Yes, ma'am.
Ma'am, I'm just sharing it so that I can check it. That's all fine.
Okay.
Ma'am, there is a video. Can you check that video is audible also?
Audible, but voice is very low.
Ma'am, audio hasn't started yet. I'll just...
Okay. It is visible, ma'am.
Okay.
Video also playing.
What the application actually does is has a number of sensors within there. And these sensors, they take your heart rate, they take your respiratory, and they take your temperature. Then we ask you a number of questions.
Is it fine, ma'am?
Questions about
Okay.
over the last 14.
అమ్మా పూజ
మేడం చెప్పండి మేడం.
మీరు షేర్ చేయండి రమ్మా.
ఓకే.
ఒక్క 2 minutes ఈ ప్రోగ్రామ్ దించండి.
ఓకే మేడం.
అండ్ ఇంకా ఒక 2 minutes టైం ఉంది కదా, విల్ వెయిట్ ఫర్ 2 minutes. ఓకే షెరిన్?
Yes, ma'am. Fine.
After 2 minutes we will start.
Okay, ma'am. Okay.
And Shaheen Begum is our assistant professor and she is the moderator for your session. Shaheen.
Okay.
Hi, Dr. Shirin. Hi.
Hello, ma'am. Shaheen, ma'am, anytime actually when I start giving my presentation, I am totally in a flow. So I normally don't check that sometimes internet issues happens. So if there any issue, you just you can just ping me or you can just update me if there are any issues.
Yeah, sure. Yeah, definitely.
Okay.
Sure. శ్రీదేవి మేడం, మీకు మేడంది క్లియర్ గా వినిపిస్తుందా? వాయిస్ కట్ అయిందా ఇప్పుడు?
లేదు, వినబడింది. మీకు?
మీది కూడా కట్ అవుతుంది నాకు వాయిస్. నేను డెస్క్టాప్ నుంచి
ఒక్కసారి ఆడియో సెట్టింగ్స్ ఒక్కసారి చెక్ చేసుకో. నువ్వు మొన్న కూడా నువ్వు ఓట్ ఆఫ్ థ్యాంక్స్ చెప్పేటప్పుడు సగం వినబడింది, సగం వినబడలా.
బ్రేక్ అయిందంట.
అందుకనే ఒకసారి ఇనిషియల్ గానే చెక్ చేయి.
అవును, నాకు వాయిస్ కట్ అవుతుంది ఎందుకో. అది డెస్క్టాప్ నుంచి అనుకున్నాను నేను, కానీ ఇప్పుడు మొబైల్ నుంచి కనెక్ట్ అయినా కూడా అలానే వస్తుంది.
అంటే ప్రాబ్లమ్ మేబీ విత్ యువర్ హెడ్సెట్.
హెడ్సెట్ కూడా మార్చాను కదా మేడం ఇప్పుడు. అంటే మారితే అది కూడా మారింది. ఒక ఇంటర్నెట్ కనెక్టివిటీ స్టేబుల్ గా లేదు అని చూపిస్తుంది మేడం. మొబైల్లో కనెక్ట్ అయినా కూడా అలానే చూపిస్తుంది మరి.
అవునా? ఇప్పుడు మాత్రం వినబడుతుంది.
అవును, ఎవరైనా వీడియో ఆన్ చేసినా ఆఫ్ చేసినా వేరే వాళ్ళదైనా కూడా నాకు డిస్టర్బెన్స్ వస్తుంది.
ఓహో.
నేను సేఫ్ సైడ్ గా రెండిట్లో జాయిన్ అయి ఉంటాను.
ఓకే.
హెడ్సెట్ లో ఒకవైపే క్లియర్ గా వస్తుంది మేడం. ఇప్పుడు మీకు వినిపిస్తుందా నా వాయిస్?
మళ్ళీ లోగా వస్తుందిరా వాయిస్ ఇప్పుడు.
ఇందాక బాగుందా? సరే మేడం, ఉన్నదంతా సరే మేడం, ఉన్నదాంతో అడ్జస్ట్ అవుదాం మరి.
అంతే, ఇంకేం చేస్తాం. దాని వాయిస్ నువ్వు వాల్యూమ్ పెంచుకోవడానికి రాదా?
వాల్యూమ్ అందులో రావట్లేదు మేడం.
ఓకే. Sherin, shall we start?
Yes, ma'am. Yes, we can start.
Yes.
Okay. Amma Pooja, you just share the biodata slide. Amma. Chapra Shaheen.
Madam. Yes. Okay. Shall I start, madam?
You can.
Okay. So, good morning, dear participants and colleagues. Thank you so much for being here with us in this morning. So, it's my pleasure to invite Dr. Shirin Zafar, today's resource person. Today, she is going to deliver her presentation on how AI is enhancing the ability to track and fight COVID-19. Dr. Shirin is currently working as assistant professor at Jamia Hamdard University, Delhi. She is teaching various BTech and MTech courses. Madam has pursued her PhD in 2015 on optimized secure protocol from Manav Rachna University, International University. Madam has around 20 publications in the national and international journals. So her research area is soft computing, meta-heuristic algorithms, and network genetic algorithms, and big data computing and cloud computing. So coming to the achievements, Madam is editorial member, reviewer, and for various conferences. And she has reviewed around 87 papers till date. So I welcome you all for the presentation. Over to you, Madam, Dr. Shirin Zafar.
Thank you, ma'am. Thank you for the kind introduction. Okay, I'm just sharing my screen. Ma'am, is my screen all visible?
Visible, Sherin.
Do it now or sometimes later will become never. In this current pandemic scenario, I would like to tell everybody, whichever field we are belonging to, whether we are in a pharmacy field, whether in a computer science field, whether in different scenarios of life, we should all strive together for the betterment of the society. Lockdown always cannot be a solution. So we should find some solutions by using our technical expertise that we have, and collaboration in this current background plays a very important role. So you cannot say that I am belonging to a pharmacy department or I'm belonging to a medical field, so I cannot have a research on a artificial intelligence background or a machine learning scenario. So we should all work together, because nowadays everything is connected. Nowadays everything has some connection with one field or the other. So in this my presentation, I would like to focus how COVID-19 spread is being controlled and being managed by using artificial intelligence, and various types of pharmaceutical industries and healthcare workers have been benefited by using the concept of artificial intelligence and machine learning. Firstly, I would like to thank Shri Padmavati Mahila Vishwavidyalaya, Women's University, for organizing this kind of FDPs, because in this current scenario, since we all are doing these things online, we are gathering one or more information, or we are gathering some kind of specification by attending these kinds of workshops. So it's very, very important that these FDPs and workshops should be conducted, and people, at least if they are opening their laptops only for a little bit of time, they are gaining something. So it can be a very, very important scenario for research analysis. Let's start with a video which specifies how artificial intelligence and machine learning are playing a very, very important role in this COVID-19 scenario. Let's see this new technology. This is a touch-free technology, a fit-to-fly system that has been installed at the Abu Dhabi airport, which helped to specify the travelers in using the Etihad Airlines.
What it has a number of sensors within there, and these sensors, they take your heart rate, they take your respiratory, and they take your temperature. Then we ask you a number of questions, general questions about what you've been doing over the last 14 days. Once those questions have been answered, there's an algorithm that sits behind there that calculates. If we have a concern, then that will flag up a secondary assessment. One of our roaming agents will come over to assist you, and then we will conduct the secondary assessment with our company Medlink.
No.
See, it has a voice recognition system. It has motion sensors.
Because of the sensors that are involved, and you stand in front, and then screen comes up. So there's a white circle, and what you must do is have your head in there so it goes green. So once it's green, it's taking your temperature, your respiratory, and then your heart rate. Now it's asking me, do I have a cough? And I'll answer no. And we also have head tracking here. So with head tracking, I look down to no, and the cursor will follow my head motion and go to the no and register, which is now saying that I'm fit to fly, and it will now go through to the next process, which is to drop the bag off.
So we have seen in this video how artificial intelligence is being utilized in various types of airports, and this technology is in a trial phase, but many of the trial versions have been proved very successful, and it is playing a very, very important role for our war or fight against COVID-19. Normally what I do, I have a connectivity with my audiences. So I normally have a rapid fire with my audiences. So see how much the people are connected with the speaker. So I think let's have some answers from the audiences. So if you know anything about artificial intelligence or machine learning, let's know how much we have. So let's see how the audiences are connected with this particular session. So let's have some answers. So let's go with the first question. The intelligence displayed by humans and other animals. Do anybody know? Any answer?
Audience,
can anybody give me some answers? The intelligence displayed by humans and other animals. Anybody? Just a guesswork only. When we are talking about, Vaishali says deep learning. Anybody? Anything? Any other answer?
Artificial intelligence?
Yes, natural intelligence. Yes. Yes, sir. It's artificial intelligence, or when artificial intelligence started, it was under the concept of natural intelligence. So it is all revert as natural intelligence, because artificial intelligence is also a part of natural intelligence only. Yes. So I got an answer from whom? Can I have the name? Okay.
Good morning, ma'am. It's Amaresh. Amaresh from Bhubaneswar, Odisha.
Okay. Thank you, sir. Amaresh, sir, you were very close. Thank you, sir. Okay. Now, second one. Any device that perceives its environment and takes actions that maximize its chance of success at some goal terms. Achieving the goals in artificial intelligence. Any device that we use. You just saw in the video also. If the audiences saw the video, you saw that device in the video also. Anybody, any answer? If you saw the video, you saw that device also. Yes, Jaypreeti ma'am, it's sensor, or they are also called as intelligent agents. Yes, Jaypreeti and Prashant, you are totally correct. They are also called as sensor or intelligent agents. Correct, totally correct. Okay, so we have a very connected audience. Now, third question. Anybody knows in which year artificial intelligence was found as an academic discipline? Can anybody have some guesses for that? In which year artificial intelligence was found as an academic discipline? Very close. Nisha Rani ma'am is very close. Okay. 1953, very close you are. Anybody, any other guess? Any other guess? So we'll go with Nisha Rani ma'am. It's 1956, ma'am. It's 1950. Yes, it's 1956. Correct. Totally correct. So we have just a very connected audience. Okay. Now, can anybody tell me any tool used in AI? Can anybody tell me any tool used in AI? Any knowledge of any tool used in AI? Okay, computers. Okay, let's have some more better answers. So what I'll do, okay, Python is one. What I'll do, I'll come with question number 5 till 10 after this session. We have 1 to 5 are more connected with the video and all. So we are coming with question number 5 till 10 after the completion of this session. Okay. So very good. Okay. So artificial intelligence tools we'll look at, and we'll come back to these particular questions after the session. So we'll have more answers, but I think the audiences are very much connected. That's so good. So I'm starting with this particular thing. So, okay. Now what we see, let's see this particular graph. This told that the graph told that when we have an uncontrolled transmission of the COVID-19, when we are using some controls and when the capacity of the healthcare system, this graph could come down. So in this particular scenario, when in December it was detected, and in March when there was lockdown being done, the main goal was that by using some controls and by using the capacity of the healthcare system, this graph could come down. Lockdown, as I told you, could not be every time the solution for controlling COVID-19. We need to use some controls, and what's so good that those controls can come with the specification of artificial intelligence and machine learning. Now, what is actually artificial intelligence? If we are talking about any technique that is mimicking human intelligence, just yes. Is somebody speaking something? So now what we are coming to the definition of artificial intelligence. So when it is the definition not only for computer science people, it is a definition for everybody. And what that definition sees, that any technique that is mimicking human behavior or human intelligence by using some rules, by using some definitions. And when we are going towards its subset, and the subset of artificial intelligence is machine learning, and machine learning uses more and more experiences. When that human mimicking behavior or intelligence is added with more and more experiences, or which is added with more and more scenarios of different technologies, then it is called as machine learning. And when that subset of machine learning is being utilized by using multi layers of neural network scenarios, image recognitions, and other kind of algorithms, then those vast amount of data specifies the concept of deep learning. So if people say that I am working on deep learning, but I have no knowledge of artificial intelligence and machine learning, this is a myth. All artificial intelligence, machine learning, and deep learning are very, very bonded and connected towards each other. So as I told you, when we start our journey, the journey is of human intelligence, that is more of artificial intelligence. Then the journey goes into the scenario of more statistical techniques being utilized, more type of experiences being added, that goes Ma'am, I think there's some mic, somebody
Adil enna
from the other end.
they are having any previous history, kidney problem அந்த மாதிரி இருந்ததான்
Yes,
problem
yes.
இல்ல.
I request all the participants to unmute their systems. Pooja, can you see who is speaking? Pooja?
எதுக்கு?
Madam. Ma'am, yes, ma'am, tell me.
Madam, please go ahead. Sorry for the disturbance. There was some disturbance from the audience.
Yes, ma'am. Ma'am, am I audible now?
Yeah, you are audible.
And my screen is visible, ma'am?
Yes, it is visible.
Okay. So when we are saying that we are mimicking human intelligence, it is more kind of artificial intelligence, then it goes with a subset which has more of statistical techniques being applied. It is more of experiences being added. Then it goes to the concept of deep learning. And then we have the more of neural networks being added. So all these scenarios of artificial intelligence, machine learning, and deep learning are one-to-one connected to each other. So if people say that we have some knowledge of AI, then it's very much easy that if you have knowledge of AI, then you can go towards the learning of machine learning and deep learning. Now, people usually thought that artificial intelligence is future, but no, artificial intelligence is here now. When we can use the robots, when you can use the various types of driverless cars, and when you can use the machines to sense, the comprehend, learn, and act, then you are into the world of artificial intelligence. See, you have artificial intelligence being developed for surveillance scenario. So this is a human brain mimicking, and this is AI technology which is used for high level surveillance system. So these type of technologies are also being utilized in many of the countries, including India, for surveillances and for high security scenarios. Now, there is a race going on between China and USA of who will win the race of the artificial intelligence. But I can tell you that India is not very far behind. Around 12 to 14 startups have been built in India during this current pandemic scenario, all using the concept of artificial intelligence and machine learning, and all benefiting the society for COVID-19 fight. So this race, India is also a part of it, but the two main these scenarios and two main countries who are undergoing this fight of AI is USA and China. Now, as I told you, the work has already been started. So the companies like the AWS, they are using the COVID-19 scenarios for building COVID-19 diagnostic system, and they have already launched the COVID-19 search. And this COVID-19 search is very important for speeding up the research and the treatment. So this is one of the French startup, that is Clive.io, and they are utilizing one of the most important COVID-19 search, and this is very, very helpful for the society for the research and treatment, mainly when people are very much affected during this coronavirus pandemic situation. So now how artificial intelligence is playing a very, very important role, as I told you, the intelligence of artificial knowledge is using in the disease surveillance, in tracking the hotspots, in accelerating the diagnosis, in detecting the infections. And these are something which you are naming, and they can be so many applications. I'll show you in the upcoming slides also, where these artificial intelligence and machine learning scenarios are used, and they are one of the stepping stone, and they are the changing scenarios in different sectors, whether it's management, whether they are fintech, whether it's healthcare, and and disease diagnosis, they are proving to be a very, very important scenarios against a greatest aid against this pandemic situation. So as I told you that whenever we are using the concept of machine learning, natural language processing, the facial recognition, data analytics, deep learning, all of the things are a part of artificial intelligence journey, and it is like a ocean, and that ocean is using the industries like the healthcare and all scenarios for fighting against a pandemic also, and previously also they were utilized, but in this scenario they can be very, very important for managing the various tasks very effectively. Now, code versus COVID-19, there is an organization which is called as the Vadhvani AI. They have already started a cuff against COVID, and they are working with the Stanford University. Now, what they are doing, they are recognizing the sound of that cuff. And what's that the scenario? That whether the cuff is a normal cough or pneumonic cough, or whether that is cough is coming from a COVID-19 patient. So what they are saying, the sound is only used to indicate the further diagnostic, because even that sound of the cough can specify whether it's a cough of a COVID-19 patient or whether it's a cough of a person who is suffering from normal cough or pneumonic cough. So they have started their trials, and they have started this workout with the Stanford University. Now they are also collaborating with the Bill and Melinda Gates Foundation, and the important thing, they are also taking into the scenario that all this data which is coming to them, the digital privacy and the personal autonomy needs to be maintained. So they are only utilizing that particular data for recognizing the cough of the patient, and they are not using that data anywhere else to be transferring to some other agency. So what they are doing, if you look at this particular diagram, if a person is infected from a cough, so they have their connected device, and that connected device is informing to the user whether their cough, by the analysis of that the cough coming from the lungs, whether that cough is a simple cough of a pneumonic patient or whether it's a cough from a COVID-19 patient. So they are also utilizing this idea for the development of the society. So this is again an innovation which is being started, and the journey is although not completed, but they are working, and this can also be one of the greatest specification for the understanding of the COVID-19 pandemic scenario. Now, safety recruitment, many of the companies like countries like Sweden, they are utilizing the robots for their recruitment. So no human intervention is required there. So what they are doing, they are recruiting people and recruiting candidates all with the help of safety, and again no human intervention is required, and that hiring is being done and the interviews are being conducted, and this is also being scenario of recruiting with a safety. So their AI is working as a game changer. So another again a second kind of application where your AI is used by Sweden, in which they are recruiting people by the robotic system where no human intervention is required. Then, to maintain social distancing, a social distancer, that is a safe distancer app, is being utilized in various types of supermarkets and other crowded situations. The various types of countries like Singapore, then we have the South Korea, Germany, Saudi Arabia, and other are utilizing this safe distancer app, and people have been told to download this app, and then they are taking the images, and then they are specifying the people who are not maintaining the social distancing, and they are giving some beeps and messages are being thrown to them, and they are being caught also, and they have been asked to maintain the social distancing in order to be more and more safe during this pandemic situation. So this is one of the thing again which is done with the help of artificial intelligence, and they are also taking care scared that the privacy of the customers whose images have been captured is being maintained. So that information is nowhere transmitted to any other network. So this is also being done. So remember that when we're talking about Sweden, or when we are talking about Singapore or the Stanford University, all the data that is being taken, the privacy needs to be maintained, so that artificial intelligence works only as a game changer and not as a fraud changer. Then tracking diseases at its risk. As I told you that on 31st December, when in Wuhan this COVID-19 spread was declared, then many of the organizations are utilizing. So Blue Dot, a Canada-based AI company, was first to detect the emerging risk of this particular COVID-19 pandemic. And currently what they are using the reports in various types of languages on daily basis. And what are they using? They are using the artificial intelligence mechanism to detect humans carrying those symptoms. And also they are working in collaboration with Apple and Google to develop an app, so that this spread could be controlled, and this disease in the people can be detected by various types of symptoms that are being spread all across by the patients who are suffering from COVID-19. So another application of AI. Then speeding up the diagnosis. This is again a very, very important scenario that is utilized by artificial intelligence. Cure.ai. Cure.ai is another organization. So a person known as Rizwan Malik, who was a radiologist, and who was already working in the Bolton Hospital, and he was already developing a technology of mnemonic symptoms. And so now what he is doing, he has immediately proposed his initial readings, and now he is utilizing that particular technology for COVID-19 spread also. So when we are saying that there is a heavy load on people and the staff, and how to diagnose the COVID-19 pandemic, so this particular technology will again be distinguishing patients from normal pneumonic patients and the COVID-19 symptomatic patients. So the pressure on the health workers will be reduced. So this is the work that is already started in UK. So as I telling you, this is worldwide scenario. Now I'll be coming to Indian scenario also. So India is also doing a lot of work in artificial intelligence to reduce the growth of COVID-19 pandemic. So now what the thing we need to understand, that COVID-19 spread or any other problem, this particular healthcare industries require the artificial intelligence support in order to overcome this crisis situation. And we cannot say that future will be there of AI. The AI has come, and we have to adapt, and we have to use this technology efficiently to serve the people in various types of scenarios. Now, as I told you, when we are talking about Sweden, when we are talking about Singapore, when we were talking about Boston or UK, this was a worldwide scenario. Now, India, as I told you, is no far behind the race. So the Blog.api, the Blogs.api is a T-Hub incubator startup, which is a Hyderabad-based startup, and it has employed 30 to 50 drones at a time. And they are very important when we have the scenario of the red zones or the other zones, and they are used for tracking the situations of those particular areas. So they are useful in situations where in hotspot situations, in hospitals and unsecured locations, they are all being tracked, and they are very, very important for monitoring the public places. So this is the work that is AI based drone monitoring system that is being developed by the T-Hub Incubation Center, which is a Hyderabad center called as blogsapp.ai. So this is one of the work that along with the other works that I'll be telling you that have already started, and their drones are already being installed. So what they are doing, the most efficient work that these startups are doing, the hardware they are using the older one, but they are inbuilting the artificial intelligence algorithms, and they are inbuilting the artificial intelligence techniques into it, so that this particular scenario they can be utilized very, very efficiently. Then BlueSemi is also a Hyderabad based startup, and they are using a device called Neem, and this is a device that is used to take the temperature of the people 15 centimeter away from them. And this AI based device that records the temperature without any manual intervention. So they are utilizing in self powered IoT devices for overcoming the energy consumption and efficiency solutions. And these all Neem based thermometers are utilized in various types of smart devices and other types of areas. So BlueSemi has already started its work for working or fighting in the race of COVID-19. Now we have the ByteForce. This is again an AI based computer solution which is being developed by T-Hub. Now they are using, as I told you, they are using the existing CCTV cameras and integrating the artificial intelligence algorithm into them to monitor the crowded places like the shopping malls and hospitals. And they are recording the real time video footages, and they are alerting the people who are causing any type of violations, that is safety violations. Also they are helping people and monitoring people who are not wearing the PPE kits and maintaining social distancing by using computer vision and image analysis. So ByteForce is another Indian based startup, Hyderabad based startup, which has started its work towards the fight against COVID-19. Then Cogni.Care is again an company or startup which is used to predict COVID-19 patients in real time, and it is again developed an IR thermal sensor infused with AI algorithms. And the advantage is it is used for predicting the patients, and also it is monitoring the patients the recurrence. Okay, so if this recurrence can occur, so that's monitoring is also being done of the patients who are already suffering from COVID-19. So this is again working in a two-step procedure, that is the Cogni.Care. Then we have the Inventor Robotics. It is again a Bangalore-based startup, and it is utilizing to help the doctors and the healthcare workers. The robots are being utilized which are AI enabled, and it carries out validations such as temperature checks and all things in a contactless manner, and also have video conversations with the doctors you can have. And it is deployed in around more than 10 hospitals across the city. So this work has already been started at a very good scale. Then we had the Cure.ai. The Cure.ai I told you before also. So it is a Mumbai based startup. Then you're using the X-ray screening to detect the symptoms of the COVID-19 patients. And this work has already been utilized in 40 plus cities across 7 countries. And they are using the cloud based technologies. And the important thing is that that screening is distinguishing between the patients who are suffering from COVID-19 and who are suffering from normal pneumonia. So this work has already been started, and they have utilized in various types of countries and cities all across India and other countries also. Then the Rayvision, this is again a Chennai based startup that is using the CCTV surveillance detection system for monitoring the people who are doing the violation of the face mask, the social distancing. And they are using the existing CCTV cameras and inbuilding the AI based technologies. And various industries including the manufacturing, the IT, the pharma, all are utilizing this Chennai based startup in order to develop the CCTV surveillance which is installed and which is combined with the artificial intelligence approach. Then Stacku is again a Gurgaon based startup that is again using the AI based thermal cameras, and this is called as the Jarvis cameras. Those Jarvis cameras, they can be located at a range of 100 meters and can monitor multiple people at the same time. And they are very, very helpful in crowded places like airports, railway stations, malls, etc. And this is very helpful for identification and suspects and PP monitoring, safety analytics. So this is so much interesting work that is being started up by various kind of small startups, and they are using this particular pandemic situation to work at a very large scale. Now, Terex Soft is again a T-Hub incubator that is utilizing the AI approach in the CCTV cameras, and they are also working with facial recognition, edge deployment, and real-time business analytics. So they are working and helping people, and even they are helping for the economic boost also. Then the Veda Labs. The Veda Labs is again a Gurgaon-based startup. They have installed about in 4,500 stores this AI-based CCTV cameras, and they are very, very helpful in thermal scanning and the visitor authentication. And they are again using the facial recognition technologies. So this deployment of those cameras is already being done. So when we talk about Chennai, we talk about Hyderabad, Gurgaon, the work is continuing at a very, very large scale. The Vihan Technology, that is the IIT Delhi-based startup, has also utilized the AI-based temperature monitoring system, and it is very, very helpful in monitoring social distancing, non-contact detection. So when we are talking about physical security and surveillance, this is also being maintained in all those startups which are developing where one product or the other product. So as I told you, India is no far behind in this AI race. So as I was telling you that the most important thing is that although this work is growing at a very rapid scale, but you need to take that where you are getting the data from. What happens that when people take the data from unrecognized sources, there was a Telangana based startup which had a setback. Now why it had a setback, the main issue was that that during this particular scenario, they didn't take the correct data from a correct database. Now what happened that although they wanted to distinguish between the pneumonic and the COVID-19 patients from their x-ray analysis, but since that the data they didn't get from the correct source, the analysis was predicted incorrectly. So it's a very, very important scenario when we are doing any kind of research or when we are doing any kind of analysis, that we get the data from the best and the most recognized source. Otherwise our technical work and an algorithm development will all be a failure. Now, the thing is that we are always thinking that what is weak AI and what is strong AI. All this work that I was talking about is all coming into the specification of weak AI, because we are developing chat boxes, we are utilizing the artificial intelligence in CCTV cameras and thermal recognitions. So we are just in the scenario of weak AI. But what will be strong AI? Strong AI would be like more of human-like intelligence, which will be a driverless car, which will be kind of a cognitive skill development. And when we are talking about any type of science fictions and movie like robots and other technologies, they will be more of strong AI. So there was always a fight on people that people think that AI is uncontrollable. No, the weak AI that is being utilized in the current scenario is very much controllable by humans. I know that strong AI would be a difficult thing, but with human intelligence, the control of that strong AI would also be done in some manner or the other. So you cannot stop AI being coming into the picture. You need to bring AI into the picture, but you need to have algorithms controlled on that particular scenario. Now AI is helping us in very, very important steps, and those steps can be more of you are having a more kind of less risk. You have no breaks and no human errors in the development of any kind of tool and specification. So there is no slowdown, and you have fewer cores to done in order to develop any type of artificial intelligence product. Now better forecasting and better resource distribution. And as I told you, when we are talking about the disease diagnosis and disease analysis, that is also being done in a very, very efficient manner by the artificial intelligence approaches. But each technology has some of its disadvantages also associated with it. So in Indian perspective, if you talk about, although India has started its journey very efficiently for the artificial intelligence, but still when we talking about agriculture and logistics and manufacturing, these technologies need to be embedded at a very fine scale. And the problem that artificial intelligence obviously has, it is no abstract thinking. So it's the machine. So obviously it cannot be as sensitive as humans. So this is one of the disadvantages that scientists pose. Then obviously there will be no ethics and more sloth. This is very important because people are becoming lazy and more dependent on machines. We need to make the machine dependent on us, not human dependent on machines. Then this journey of artificial intelligence would be very, very efficient. So as I told you that it's very, very important to take up artificial intelligence for sustainable growth of any kind of country and the company. But the thing is that we need to minimize the failures. We need to ensure any kind of happiness all across the globe for using these technologies, and we cannot create the things that are uncontrollable. So you need to use artificial intelligence in a very, very controllable fashion. So these initiatives needs to be adapted for any type of business development, decision making, and deploying large scale development. So these type of technologies are the important scenarios that need to take up for the development of the society. Now, when we talk about as learners, as researchers, so we have various types of tools to work on as researchers. So when we talk about, we have the Azure machine learning model. So if you are a researcher or if you are a learner who want to learn new algorithms of artificial intelligence, then you can start your journey with the Azure machine learning. It is also a cloud-based service, and it's very important that it can be used to test the machine models, the run the various types of algorithms. And even if you don't have those advanced skills also of machine learning, you can start your journey with the free version. And so the writing code is easy, but a little bit performance it has lagging when you have huge lines of code to be written. So but when you have your initial journey to be started, you can work with the Azure machine learning workflow. Then the cafe is another product or another tool that is developed in the Berkeley. And this particular tool is also very efficient for image classification. And since it is very much connected with C++, so people who have the C++ knowledge can utilize this particular tool for development of machine learning models. So it has a readable source and it has a good performance. So it's another tool, Cafe. You can utilize it for development of the machine learning models. Then we have this computational network toolkit, CNTK. It is more connected with people working on neural networks and graph performances. And since it is again a networking toolkit for artificial intelligence scenario, so you can utilize this particular tool, but it has little bit of issues with the source code and so on. But for a learner perspective, it is a good tool for development of various types of machine learning model. Then deep learning 4J, this is more suited for people who are more connected with the Java. So when you are a little bit more connected with Java scenario, then you can use this deep learning 4J, and it has more connected with the Java environment. And the important thing is that it can integrate with Hadoop and Spark. And so people who are working on Hadoop also can utilize a deep learning 4J environment for development of machine learning and your deep learning and artificial intelligence based models. Then other tools are also very important, which can be utilized, is the IBM Watson, which is also called as an answering machine. So this is also can be utilized for preparing and training and deploying your model. The main disadvantage that IBM Watson faces is that it is only available in English. So although it can be fruitful for Indian perspective, for some other countries are not utilizing it very much because of the language bar. But it is important that it can be help in business insights, and it can provide models for making decisions, and predictive analysis also it can do. Then Keras is also an open source environment which is very much utilized various type of MTech and PhD students, and people who are good in Python and who have a very good working skills with Python. So because the Keras open source is written in Python, so they can utilize for creating of their machine learning models for training and compiling and getting the predictive analysis. So this is also a Python based tool. Then we have the Pibrain. Pibrain is again a Python based tool, and it is also utilized by various types of scholars and learners for a neural network and deep learning model. And it has the algorithms for reinforcement learning also. And this is also very much a publicly accepted tool for development of machine learning model. And again, it's open source. Then Scikitlearn, it is also utilized for data mining experiences, and it is also a good visualization tool. And the advantage is, since again it is built on a Python framework, so it is working, it's better than R, but for larger data sets it's not being utilized. So again, for a learner perspective, you can use this particular tool. Then SwiftAI is also again a tool. Their GitHub page shows the direct projects where you can use the software for various types of development of machine learning model. The main advantage it has working with Mac machines, but the disadvantage is support for Linux machine is not still there, but it is in the progress. So you can use the SwiftAI also for development of your machine learning models. Then Tensorflow, this is very much utilized because it has a combination of both Python and C++. So Tensorflow is very much developed Google's product. So it's again a very trusted product, and it has lot of databases also being available, and it is again an open source library being provided for the advantages for development of various types of machine learning models. Obviously, some cons it has, that it does not support external data sets from Cafe or other tools, but people are using TensorFlow very efficiently for developing of their models. Then Theano is also a Python-based framework, and it is used for both the CPU and the GPU machines. But although not very much popular, it is not very popular due to the disadvantages of coding and some other issues. But this is also one of the framework that can be utilized for development of machine learning models. Then Torch. The Torch is also an efficient tool, but the main problem Torch has that Torch is using Lua. So Lua is a new language. So people who you need to use Torch, they need to get enhancement of Lua. The older versions, older tools that I was talking about, they were more connected with Java, Python, and C and C++. So there people didn't had to learn a new language. But if you want to use Torch, you need to learn a new language. So but it can be an advantage also, you can learn a new language. So that's why Torch can be also a good learning tool for the people who want to learn Lua. So this is again underlying a very good machine learning tool that is utilized by various learners and researchers. So all these tools play a very, very important role. And with the help of tools, as I told you, that artificial intelligence is not only working for the pharmaceutical and your disease diagnosis, it is also working with business perspective. So as I told you that in this COVID-19 pandemic, there's lot of effect on the economy also. So for economic development also, AI is proving to be a very, very helpful scenario. So as told that around 80% of executives says that AI will boost their productivity, and so is the increase in the investment in AI. So this world says that AI and business are working as a perfect fit. So for various types of organizations, AI is working in increasing their productivity, in automating their customer support, improving their efficiencies, and then there is improve in their real-time audio and video analytics, and the customer satisfaction and the customer support that is being specified is also a main perspective that artificial intelligence is focusing upon in order to develop the business scenarios. As I told you that whenever we have various types of fraud detection or recommendation system development, online searching and mining, all are a product of artificial intelligence and machine learning, which are giving a very, very boost to the society and to the economy so that business can flourish at a very fast rate. So it's very important that we need to specify then when we are talking about retail, the machine learning algorithms are helpful in predicting the customer purchases and activities so that various types of fluctuations and demands can be controlled by the business perspectives, and the business can flourish at a very fast rate. Similarly, when you have the fintech organization, the financial services, they are using the machine learning for predicting the risk. They are also using the machine learning for reacting to market trends. Even the BFSI, that is the banking and finance sector, is using this artificial intelligence for getting dealing with the various types of anomalies in the transactions and frauds. So the algorithms are help to specify the trends in the human purchases and the transactions, and that too in the real time scenarios. So when we talk about retail, when we talk about the financial services in the economy boost up, all these are using artificial intelligence to boost up their system. As I told you, this we have already been discussed that in the healthcare scenarios, whether you are using the robots or the health monitoring systems, and all the IoT devices require these types of machine learning and artificial intelligence algorithms to boost up their work. So this is the scenarios that when you talk about retail, when you talk about manufacturing, healthcare, financial services, travel, hospitality, even your energy and utilities, all are using the artificial intelligence for various types of planning, diagnostics, alerts, scheduling. So you name one work, and artificial intelligence is connected with each type of activity. So normally what happens that organizations are in little bit of they are in a dilemma how to get started with AI. So it's very, very important that you need to identify at which point in your business artificial intelligence is required. So you need to focus on those touch points, and you need to find the best algorithmic solutions for decision making. And necessary training also is very, very important for the employees, because otherwise there can be some issues in the product development and bringing that product into the market, and customer satisfaction can be affected if you are not skilled in the terms of artificial intelligence tool development. So when you talk about that, AI is a very important scenario. So AI, the for business imagination and implementation, it's very important that we should accept AI and control it. It's very, very important that the risk of business can be controlling, and you cannot ignore AI in any type of scenario. So AI need to be accepted, but AI needs to be controlled. So then the thing is that it's very, very important, as I told you, that you need to determine where your business or where your product requires AI. So those touch points and the game changing events need to be captured, and you need to specify the questions, you need to specify the problems, and you need to use the small algorithms for getting that predictive analysis being done. So the determining for your AI for your business works as the first scenario. Then it's very, very important that you have to have an application being developed that is being suited for your problem, and you need to prioritize, you need to specify that how these algorithms can help in automating the decision and can help in your decision augmentation, and how can this be a business value product. So you cannot say that at every point you can apply AI. You need to identify those touch points so that you get the maximum impact of artificial intelligence. Then the culture for AI is very important. Nowadays people keep their back steps, say no, I am okay, I can do manually. But no, you need to adapt it. You need to specify that that AI is there. You need to have some technical sessions with your employees, with the startups being developed. And as I told you, that AI is need to specify that the current updations happens, and it is not an on-off project. You have to, once you have taken it, you have to adapt it and work according to your scenarios for your business development. And it's very, very important that you have to invest for the long term in data for AI, understanding and the culture and the evaluation the data. As I told you, getting wrong data could be just a loss of business. So this understanding the culture for AI and getting the best fit algorithm for AI is the ultimate solution for any type of future development. And as I told you, training, getting the right talent, also you can have some type of workshops and skilling the people. Even the data scientists and data engineers can be employed for the organizations for getting a best perspective of this particular AI based technology. So when you are talking about your revolution with the healthcare analytics, AI is already into the business and doing a lot of work. So as I told you that in America, people don't like visiting the doctors. Even in Japan and Germany, they have those AI apps and robots being connected, and they get their device advice from the doctors without visiting them. And in the corona perspective, this could be very much efficient, that you don't have to go out and you don't have to meet people. You can directly get the advice and get the best possible solution for your health related issues. So when we're talking about your drug discovery, AI and is working at a very, very efficient way, even in predictive modeling and monitoring of the live data. So this is the helpful specification on development of drugs. And the thing is that since the coronavirus is more of a mutative process, then the issue is coming for the development of the vaccine. But AI is also working in the predictive analysis and providing lot of researching perspective for the scientists. So when you're talking about the diagnostics, the scientists at the Stanford institution are creating algorithms that can visually diagnose a person potential cancer cells using a database of 130,000 skin diseases images. So as I told you, when you're talking about your Stanford University, the IBM Watson, the Google Deep Mind, the Boston-based biopharma industries, they all are utilizing this AI technology for better therapeutic treatments, better treatments for degeneration and cognitive, the tumor sequencing, and even the Oxford P1 vital predictive response to depression treatment predict. As we have seen in this current scenario, the depression is also cause of lot of deaths. So this emotional connectivity, and this is also being developed for diagnosing the depression and providing treatment for the people who are suffering from depression. So this work is working, this AI is working in a very, very effective manner in various types of your diagnosis, even your skin diagnosis, your cancer diagnosis, and your depression treatment also. And as I told you that AI is working as a telemedicine, and this telemedicine is aiding for more and more precise diagnosis, for more better treatments by using your pattern recognition, and also various type of logistic challenges and administrative headaches are also being controlled by using the AI technology. So AI is also utilized for conversational analytics. You have Siri and you have another types of supportive conversational analysis that helps in people for doing the various types of tasks. So AI, as I told you, is one of the most efficient technology for businesses as well as your healthcare specification. So according to a report by BofA Merrill Lynch, robots and AI solutions will grow up to about $153 billion by 2020. So that's a very important thing, that you need to specify a checklist for AI development. You need to do lesser mistakes that will grow when you have little bit experience of it, a limited judgment, a high volume, repetitive, that you need to have a predictive analysis, and you need to have a pattern being followed in order to specify a good predictive analysis for your AI based algorithms. So as I told you that AI has already started a lot of automation in various types of decision making scenarios. Even your chat boxes and even your transactional flows, they are utilizing in marketing, sales, and customer service for producing best and better solutions for the companies as well as for the customers. So very, very important scenario, as I told you, that identifying the problem and identifying that source, and then developing the AI based solution, and then providing your implementation and action. Because unless and until your problem is being identified and you are getting the data from a recognized source, then if those two things are not doing correctly, then your AI based solution and implementation and action can be a failure. So these things need to be judged in order to get a best possible solution. So when you are talking about the key drivers for creating the exponential growth of AI, so the advantage is that these advanced algorithms and the workforce, they are also playing a very, very important role. Because unless and until those algorithms and the workforce, that is a skilled workforce, is being utilized, your experiences for AI development could be, again, as I told you, will be a failure. So when you talk about your predictive analysis and your man and machine connectivity, so organizations are using this machine learning scenarios for various types of game changers technologies, for various type of analytic developments. So as I told you, when you're talking about your Alexa, your Siri, your Watson virtual agent, all are creations of artificial intelligence, and they are also developing trust and maintaining the privacy of the user, so that the artificial intelligence could be an adaptable technology for future also. Now people, what happens that normally some people say that I am not very good in coding, so I cannot use artificial intelligence. But I can tell you, this machine learning and AI can also be very much benefited by using the visualization tools. So if you are not very well versed with coding, you can go with the visualization tool Tableau is the best fit example, because it has just the drag and drop, and it is a visualization tool that can accept data from any source. You say a source from Hadoop database, you say a source from Excel, you say a source from PDF, you say a source from any type of database, you are accepting that data in Hadoop, and it helps in a few clicks, no coding required, just the smart dashboards and drag and drops, and you can develop your predictive analysis solution by using the most trusted visualization tool. Even the Gartner is the one of the most trusted business intelligence solution providing company. They told that Tableau is one of the best fit visualization tool for generating predictive analysis solutions. But although Tableau is 14 days free version, but if you think that Tableau can be very supportive for your organization, you can use this Tableau version. It has some paid versions, and it has also a 1 year free version for students. So students get that 1 year free version by connecting to Tableau and submitting their documents that they are student of a particular organization, and they get 1 year Tableau subscription free. Then ClickView was started before Tableau. It is also one of the good visualization tool, and again you don't require those programming skill. But when you compare ClickView and Tableau has gained more public experiences, because it's more interface is more attractive. So ClickView is now converting his interface like to Tableau, so you can use ClickView also, but when you are comparing, Tableau is much more acceptable. Then there's another tool called as a Chartbox is also a visualization tool that helps in visualization of chart building development of websites and social media scenarios. So they are also working in a very efficient manner. So this is also can be utilized as one of the visualization tool. Then Power BI is also one of the visualization tool that has a free version associated with it, and it is simple to integrate the API in the Byte platform. So it offers web, mobile, and desktop support. It is also in a very effective dashboard and real-time data solutions for the Power BI scenario. Then raw is again one of the visualization tool that helps in visualization from various types of vector files and png files also. So your any data from excel or spreadsheet can be utilized for developing predictive analysis and visualization solutions. So these are the tools which are more suited for people who stay away from little bit of coding, so they can use these visualization tools for developing models and developing predictive analysis scenarios. Now the thing is that since if you want to work more for visualization, you can go with the developer tools also. But this is my personal recommendation that if you are going with visualization, first you need to go with the audience tools, that is Tableau and ClickView. And if you are efficiently working with them, then you can go with the developer tools like the Google Charts and Fusion Charts, because there your programming skills is required and you have your Java and PHP and various types of programming environments need to be utilized to use these your developer tools, which are the d3.js, which also requires a JavaScript environment. Then we have the charges.js, which also require the CSS and JavaScript functions. So when you are totally into the visualization tool, you can go with the audience tool. But if you are need to use those developer tools, you need to have some JavaScript and these CSS knowledge, then only you can utilize them for the development of your visualization scenarios. Now, RPA, that is a robotic process automation, is also one of the field that is being inspired from artificial intelligence and machine learning for developing of various types of tasks of query processing and calculations and maintaining of records and transactions. So if you talk about the RPA work, our RPA work can be simple of reading an email, opening an Excel attachment, then entering data from Excel in the ERP platform and deciding whether the employee need to be escalated or no. So this is a simple RPA, one of the scenario of RPA that is being utilized. And as according to the current trends, the robotic process automation and the robotic desktop automation, their annual expenditure and their annual growth in 2020-22 would be very much higher scale. So when you are going in 2016 to 2020-22, the jump of the RPA will be a very effective specification. So it's again the work of all AI and ML. So this is also one of the application that is being utilized for the development of the society specification. Then people say that I am working on data science. So it's very, very important to see that this journey of data science is nowhere away from artificial intelligence and machine learning, because this one journey of data science has the fundamentals. It has the statistics, that's your maths. It has the programming, that is your Python or C or C++ or your Java. Then it has machine learning. As I told you, machine learning is also a combination of these languages. Then visualization, the tools that I have told you, which can be your audience or which can be your developer tools. Big data, very, very important, because this data needs to be generated, needs to be analyzed to get the correct predictive analysis. Then when you are talking about your problem solving approaches, these are the combination of your data science roadmap. So your data science journey is also connected with your machine learning and artificial intelligence journey. As I told you in the starting only, you cannot be saying that I am doing this, I cannot do that. But now we are in this particular society and scenario that collaborative learning and collaborative using of technologies is a very, very important role. So with the software industry, as I told you, the software industry, whether you are using the blockchain, whether using your game designing, your big data, analytics, artificial intelligence, all are using your machine learning in one or other manner. So the software industry, in terms of student perspectives, can open new rooms, open new development scenarios for our students also. So people normally, students ask, ma'am, which field I should adopt to? So these are so many fields being open to you, so you can go and have a collaborative learning in any field that is best suited for your research and development. So from Jamia Hamdard, I can say that in this COVID-19 pandemic, although we are stuck somewhere, but you can utilize this time for more of technical learning and expertise in one manner or the other. And this Atal FDPs are providing us the scenarios for enriching the learning experiences. So these are my mail IDs. You can connect to any of my mail IDs, and some of my papers are also being used there, specified there. So we can have some collaborative learning and technical analysis in any of your or my work that we can work for the development of the society. So thank you, everybody. Thank you for your support. And if any questions you want to ask, I am open with that.
Pooja, is there any questions?
Box options as of now.
You are not audible.
You can type your questions in the chat box.
No questions as of now, ma'am.
Okay, there were no questions on chat box.
If you can, if your voice is not audible, you can
Okay,
type.
Sharon.
Yes, ma'am.
One question from my side.
Unmute themselves.
Yes, ma'am.
So which mission to you told so many tools for machine learning?
Yes, ma'am.
So which tool is suitable for our pharmacy background?
Ma'am,
pharmacy people.
Ma'am, if you're working with currently I am also working with the COVID-19 database, and we are also getting our data from Jamia Hamdard, the pharmacy department only. So ma'am, we are working with the Keras tool. So that tool is very, very important for and very.
Will you type that? Will you type that one?
Keras. Keras.
Keras.
Yes, ma'am.
So this tool we have to learn.
Ma'am, actually the learning, the most important thing is that nowadays we have the videos. Okay, so I can share some links for the videos also. So just a 1 or 2 days journey of the videos can help you in little bit understanding of Keras. Then ma'am, there is a GitHub library. So in the GitHub library, you have a lot of code which is already being displayed and which is already being available. So ma'am, the best thing is that what you can do first, as we also do, because Keras was new for us also. So ma'am, what we did, we utilized the old code first, run it, and what we did, ma'am, we played with it. So we tested it, and how we tested it, we didn't use any of a new code. We didn't do any new code journey. We just run the old code, and then what we did, we tested it for their data only, and then we tested for our data. So it was a very good learning experience, ma'am.
Okay. So will you share that one? Okay.
Yes, ma'am. Yes, ma'am. I'll share that links also.
Okay.
It may be helpful.
Hello.
There's a question from Sindhu, ma'am.
Yes.
For method development in pharma analysis, which tool can we use?
As I told you, ma'am, Keras is one of you can use Tensorflow also, you can use Keras also. And ma'am, the algorithm, that is a decision tree. We have utilized the decision tree algorithm for our development. But ma'am, this is not only limited. There are number of algorithms that are available. And the best, ma'am, in this phase, the best thing is that there's so many code already available in the GitHub. So the advantage that we take and our PhD students and our MTech students do, they run that code only, and once they are well versed with 2-3 codings, then they develop small changes. Like there's a playing scenario, they change 2 lines, then they change 3 lines, and then they are able to generate a new code. Because a problem when occurs when you start writing the new code, and with a new tool you feel that you are lost, because that's a new tool and new code, so you feel lost. So this I tell to every of my students and our project development team also, because we are using that tool for project development for Jamia Hamdard only. So the thing is that you need to first play with it, just change 2-3 lines, and then start development of your new code.
So for that purpose, first we had to learn the language now, how to do that What is the basic language that we need to know?
Ma'am, the
No,
thing
no, no,
is that,
language learning.
ma'am, the thing is that they are more connected with Python and C++ and Java. So what's the important thing is that if any of the student of computer science you can connect in your project team, because nowadays even the BTech students are very good in this Python development, and they are very good in their various types of, although they may be not very much development in Keras, but they are very good developers in Python and C++. So in our team we also have those BTech students and MTech students. So what help them, they have, and the advantage them, and they are young minds, they have those ideas, so they can change that code very easily. So that's the advantage we have.
Okay, so we can take the engineering people.
Yes, ma'am. Any computer science students you can connect in your team, and especially the BTech final year students, they are very, very young minds, and they like to play with the code. The PhD students don't like, they have little bit more of a research scenario, but the BTech students are very much enthusiastic in doing those changes.
Okay. Okay, definitely we will do that.
Yes, ma'am.
Shaheen, anything else?
Yeah, no, madam, because it is really a new area for us. Maybe it's not new, the term is maybe new, but with this FDP, we came to know some of the things. Now, madam is telling decision tree, with which the means the algorithms and all. When we practice, I think we will come to know what is the fruit of the tool. For example, if we are using Keras, what are the particular areas? For example, in our drug development process, we have drug target identification and validation on all these. Where exactly this tool fits, we don't know. We have to find out that thing. And once,
that's why, as I told you, that a team collaborative work is very important. We cannot say that science people like pharmaceutical only, there's a pharmaceutical team. No, ma'am, we have our doctors connected with that, because they are very much effective in they give us these analysis without any type of predictions. So we have a doctor team, we have a pharmaceutical team, and we have the computer science, and even the IoT, that is electronic students are there. So they help in electronic and IoT developments.
So such type of collaborations definitely will give us fruitful results. Otherwise, sitting alone definitely it is not,
whether your institute is willing to MOU with our institute also?
Yes, ma'am, we can have a talk on that. That's no issues. We can have a talk on that, and you can mail me important scenarios. I'll connect you with my dean, and they'll be very much because we require those pharmaceutical background people, because as you know, ma'am, Jamia Hamdard is #1 in pharmacy in the current ranking. So yes, ma'am. So we are very much open with new ideas.
Okay.
And ma'am, one thing I would like to connect with the AICT anytime, a suggestion from the AICT, like Atal is a very good platform. We can have these FDPs in terms of like project development FDPs, in which there could be 30 to 40 days, and people could do the project development in those FDPs. So that would be very, very helpful, ma'am.
Okay, it's a good proposal.
Yes, ma'am. And they'll get a new product after those 40 days or 30 days.
Oh, yes. It's good. Nice answer.
Yes, ma'am.
Okay.
Madam, now if there are no questions, can we proceed to vote of thanks, madam?
Yes, you can.
Yeah. So it's my pleasant duty to propose a vote of thanks to Dr. Shireen Zafar. Dr. Shireen, the presentation was very informative, elaborative, and very clear. You were so much energetic during all the time. It was totally 1 hour 10 minutes, I think. You have shown a lot of interest. We got to know that Indians are not behind in using AI to fight against COVID-19 with so many startups in India. We got to know what is the connectivity between deep learning, machine worldwide.
You are disconnected.
How worldwide this AI technology is useful in maintaining social distancing, in diagnosing the transmission of the disease, and also diagnosing and also using various sensors like face recognition and voice recognition, to fight against or to maintain this situation. Dr. Shirin has provided us the information regarding what are the various tools available for the academicians and also for the business, and what are the various elaborative applications of AI in business, academics, and all other important things. So in healthcare system also. So how to get started with AI also, we got some clarity, and how what are the other applications, potential applications of AI, we also got to know. So thank you so much for sharing your valuable contribution to our FDP madam, and thank you for sharing your valuable time with us. Once again, on behalf of our organization and our institute, I would like to thank you for sharing your time with us. Thank you so much, madam.
Thank you, ma'am. Thanks a lot, ma'am.
Thank you, Sherin, for accepting our invitation and for your elaborative and very, very informative session. So it is, although it is very new to for us, but it is very interesting. Okay, thank you. Thank you very much.
Thank
Okay,
you, ma'am.
we
We
will
had a
meet
very
once again and we will mail you for your MOU. We will talk with our dean and other things.
Yes, ma'am.
And we will mail you. And some of the participants are also very much interested to interact with you, okay, personally, and they would like to call you for their college. So they need your contact details. Please post in the chat box. They will take and they will call you.
Yeah.
Okay.
Otherwise, can ma'am let us share the PowerPoint, because in the last slide we
You
have
just upload in the chat box. It is sufficient, because I'm unable to upload to mails of those participants.
Ma'am, the thing is that it is more than 25 MB, so it's not getting uploaded on the chat box. I tried it.
Oh, okay, okay.
I'll
Then
again
you just mail to me. I'll WhatsApp them. Okay.
Yes, ma'am.
Okay. Thank you, Sharon. Thank you very much.
And we had a very connected audiences, and I would like to thank you all for giving me this opportunity. And ma'am, in this pandemic situation, I have given around 30 plus these FDPs. And the main thing is that I started my journey not for any type of financial support, but for helping the society. And this is the main.
Thank you. So your motto is very good. Okay. Thank you. Thank you very much. Okay.
Ma'am, just a small announcement, ma'am.
From whom?
I just have given some names in the WhatsApp group, ma'am. So
Okay.
I would like to request all the participants to check those names and give their full name so as to mark attendance for the remaining days.
Okay, since you are logged into Zoom through your phone, not by your registered name, so it became very, very difficult for us to make the attendance of you or upload your attendance in ACT portal. So due to that reason, please kindly, whose phone number or whose phone name is that registered, corresponding registered participant. So kindly within 15 minutes you post it. Okay. And the next session we will start within 10 minutes. By 11:30 we will start. Okay. We'll take a break for 10 minutes. Okay. Ma Swati. Huh?
Bye, madam.
Bye, bye, Shaheen. Thank you very much. Okay.
Okay, ma'am. Bye.
Ma Swati, 10 minutes. Okay. Amma, Pooja, madam no
Program schedule పెట్టమనండి
చేయండి.
మేడం. ప్రోగ్రామ్ షెడ్యూల్ పెట్టమనండి మేడం. ప్రోగ్రామ్ షెడ్యూల్.
మేడం స్వాతి మేడం ఇప్పుడు ఒకసారి ట్రై చేయండి మేడం.
స్వాతి ఒకసారి అన్మ్యూట్ చేసుకోరా. అయిందా?
Yes, ma'am.
Okay. Okay, just talk. Swati.
అన్మ్యూట్ చేయమంటారా? ఓకే మేడం.
అయిపోయింది.
ఓకే ఓకే. గుడ్ మార్నింగ్ మేడం. వస్తుంది అనుకుంటున్నా.
ప్రోగ్రామ్ షెడ్యూల్ పెట్టండి మేడం. స్లైడ్
పెట్టేసేషన్ పెడుతుంది. She is sharing. So within 10 minutes we will start the class. Okay, we'll take a break for 10 minutes. Swati.
ఓకే మేడం. ఓకే మేడం.
Okay.
Swati, head of insights of molecular mechanisms of Mani Amma. today 6th November. And 1 year later కదా. స్వాతి బాగుంది కదా టాపిక్.
బాగానే ఉంది మేడం. ఇన్సైట్ ఇంటూ ద మాలిక్యులర్ మెకానిజం ఆఫ్ ప్రోటీన్ లైగండ్ ఇంటరాక్షన్స్.
ప్రోటీన్ లైడింగ్. నేను కాలేజ్కి వెళ్ళే వద్దాం అనుకుంటే
వెళ్ళొచ్చిన అదే జస్ట్ ఇప్పుడే కూర్చున్నా ఇక్కడ.
నేను నాకు 10 కి స్టార్ట్ అయింది కదా.
యూట్యూబ్ లో లైవ్ స్ట్రీమ్ అవుతోంది మేడం.
ఓకే ఓకే
ఓకే.
అమ్మా పూజ వాళ్ళు చాట్ బాక్స్ లో చేస్తున్నట్టున్నారు. యు జస్ట్ నోట్ డౌన్ దోస్ ఫోన్ నెంబర్స్ అగైనెస్ట్ దేర్ రిజిస్టర్డ్ నేమ్స్.
ఓకే మేడం.
ఏం పెట్టిన అవసరం లా వాట్సాప్ లోనే మెసేజ్
షణ్ముఖం. Please check the WhatsApp group. Only those people with device names that I have mentioned, only those can respond. Others are not required. Please check the WhatsApp group once. I have given the list of participants who have given device names. Only those people have to respond. Thank you.
అమ్మా పూజ సార్ జాయిన్ అయినట్టున్నాడు చూడు కోహోస్ట్ చేయండి ఒకసారి.
Hello.
Hello. Hello. Good morning, sir.
Morning, Dr. Swati Garu.
Good
Can you hear
morning.
my voice?
Okay, sir.
Yeah, it's fine. Excellent.
Okay. Welcome to our.
Good morning, sir.
Good morning, ma'am. How are you?
Fine. How are you?
Yeah, good. Thank you.
Okay. Now your voice is audible.
Yeah, it's fine, right?
Okay. Shall we start, Swati?
Yes, madam.
Yep.
Huh? Okay. Sir, shall we?
Yes, please.
Oh. Okay. Pooja, share the slide, ra?
Researchers, scientists, and colleagues, and my dear students, good afternoon to all. I am Dr. K. Swati, Associate Professor, Department of IPT, SPMVV. I am pleased to be here a moderator. I welcome to Dr. Babajan Banganpalli, Department of Genetic Medicine, King Abdulaziz University. I welcome to all the participants. I am so delighted to see here. I thank the coordinator, Professor Sridevi Madam, for keeping this session, which is a very important part of our drug designing. That is insights into the molecular mechanism of protein ligand interactions by molecular docking. So it's the time to introduce Dr. Babajan, and he is a researcher. His research interest is in clinical bioinformatics, computational biology, and predictive genomic medicine. He published more than 60 peer-reviewed and invited publications, books, and book chapters. He is a member of editorial boards and program committees, and he acted as a speaker in different international journals and conferences, and invited presentations and international workshops and conferences. He obtained multiple grants funded as an investigator and co-investigator. He has more than 354 citations. His H-index is 11 and the I-10 index is 12. He achieved more than 3 honors and awards. He awarded Bioinformatics Young Scientist Award 2018, Venus International Research Foundation, India. He completed his Dr. D.S. Kotari postdoctoral fellowship from University Grants Commission, UGC, Government of India, New Delhi, India. He was a CSR SRF fellowship from CSR HRDG for graduate program, and he had one patent, that is, the title is synergistic formulation for the treatment of diabetes, and he is a member in European Society of Human Genetics, Society of Biological Chemists, Indian Science Congress Association, Asia Pacific Chemical, Biological and Environmental Engineering Society. So I invite Dr. Babajan, so please take over the session, sir.
Thank you so much. So can you see my screen?
Yes, sir.
Okay, so I think I hope my screen. Okay, so here, good morning to all the participants for this conference. I hope you all are safe and healthy in this tough situation, really bad situation now. And thank you so much, Dr. Swati and the coordinators for inviting. I would like to extend my thanks to Dr. Professor Sridevi Garu and Varalakshmi and other organizing committee members who invited me to this faculty development program on AI drug discovery. So today I'm going to give a demo classes, not a theoretical classes for you, a demo class on the molecular docking method using the Autodoc Vina. So Autodoc and Autodoc Mina, maybe you heard, most of you, I think, heard about these tools. This is a widely very popular tool, very highly cited free academic tool, which is helpful for us to perform the protein ligand interaction study. So before I gave some demonstration about this Autodoc Vina, how we can use this data, how to install this one, I just want to give some basic introduction to it. So then we can start with the actual demo section. So why we have to do molecular docking? So basic questions to comes to every one of us. So why we have to do the molecular docking? So basically, molecular docking is helpful to perform or helpful to understand the molecular mechanisms in between the ligand and receptor molecule. So this will help us to find what are the ligand binding sites are there, what are the active amino acids are there, active site amino acids are there, and what is the cavities or active cavities are there in the receptor molecules. And these findings highly helpful for us in drug development procedure. Okay, so this molecular docking method is one of the major approach in computer aided drug designing. Maybe last few days you're hearing so much information about the CAD and artificial intelligence, all these informations. So without touching a test tube, we can do the multi-dimensional analysis by without touching a single test tube, we can do a multi-dimensional analysis on drugs and its targets. Okay, so I don't want to give too much theoretical content in this session. So I means we have to be more focused on the demo part. But I just want to give you how I use this computer-aided drug designing during initial phases of my research. Actually, now I'm the clinical bioinformatician. I'm working more towards the genetics terms. So when I started my research, at the time I started with the drugs and proteins. So I built my research with the computer-aided drug designing, and I just moved to that session into the lab one. So I just want to give you how I use the CAD approach during initial phases of my research career. Okay, so here, so I started with the compound called as a resveratrol. So maybe you are the most of you as a pharmaceutical people, you heard about this compound. This compound is having a lot of anti-cancer properties. So much of anti-cancer property. One of the best drug for the cancer, natural compound. It has been extracted from the red grapes, and also it is very rich in red wine. So here, as I told you, it has so much of anti-cancer properties there for this drug, for this resveratrol compound. But the problem with this resveratrol is it consists of free hydroxyl group. When it enters into the circulatory system, so it automatically rapidly it get metabolized by sulfonation and glucoiodination process. So automatically the half-life or bioavailability of the resveratrol is very low when it enters into our body. So here, I think within 8 minutes, it is getting degraded. It is not reaching it to target site, or it is not showing that much of anti-cancer potent due to frequent degradation of this resveratrol molecule. So we thought, okay, so let us protect this group, hydroxyl group. So we just assume that if we are able to protect this one, we can extend the bioavailability of this resveratrol molecule. Okay, so we went to organic synthesis lab. They said, okay, so they can synthesize a few compound, but they are not sure whether they are going to be active in biologically or not. Then it came to conclusion, so we have to use some computational method to design a library of a compound to check some physical properties of the compound. Then we can validate them in a lab. So we approached to the CAD, so computer aided drug designing. So we added different type of linkers. We added different type of linkers to this hydroxyl group. So by using different tools like EM Campbell, chemical EMBL database and Campbell database. So we retrieve the different type of linkers from the databases and known linkers from databases. We added all these linker. So we modified these groups. We added all those linkers to these protective groups. So we created nearly 10,000 library of resveratrol analogs. And to filter down, we have used different approaches like QSS studies and physiochemical properties like Lipinski rule of 5. Maybe you all know about that one, like molecular weight, topological surface area, log p values, and hydrogen acceptor donors. And also we did this solvent accessibility, sorry, solubility. And we also use this admitox, absorption, digestion, metabolism, excretion, and toxicity also we did. So overall, by doing all this analysis, we finally come to the number of 1250 resveratrol analogs, which is just like a drug. It is following like a drug likeness property, which is having a drug likeness property compared to resveratrol. It's showing a best solubility and good topological surface area and very nice admitox properties. Okay, so we narrow down 10,000 library of compound to 1250, and further down, we perform the molecular docking. So it is already proved that resveratrol major target is a nuclear fragment kappa B, NFKB. So NFKB is one of the transcription factor in cancer states. The expression of NFKB is very high, and due to expression of high expression, it is dysregulate or it create absent in apoptotic pathways. Okay, it adheres the apoptotic pathways. So here, if the resveratrol binds to this NFKB, it is going to suppress the action of the NFKB or inhibit the action of the FKB, and reactivation of apoptosis will be takes place. That's a major mechanism. It's already proved one. So what we did is we selected this target and we performed the molecular docking, and we identified nearly more than 850 compounds, 800 resveratrol 50 compound, which is having a very high binding affinity. Okay, very high binding affinity with the nuclear fragment kappa B. Okay, so then we filtered down nearly 850 compound. Again, we performed the one more step, molecular dynamics simulation. We use a GROMACS tool, and by using the GROMACS tool, uh, here we in the solvent system, we want to check how much stable the interactions are there, how much stable interactions are there. So we again, by using these terms, we narrow down to 85 analog. Finally we end up with the 85 analogs. These 85 analogs we suggested for the organic synthesis with the help of the chemistry people. Fortunately, unfortunately, or fortunately, out of the 85, we are able to come up with the only 30 to 35 compounds, I think. So 35 compounds, then we again performed the in vitro studies. We identified all the 35 compound showing a very good results in vitro study. We performed the MTT assay, like a cell toxic assay, fragmentation assay, DNA fragmentation assays, apoptosis assays, and also we performed this target specific assay also we perform. So fortunately for us, we come to the very good results. So what I'm trying to tell you is, now we can see here from the 10,000 compound, we are able to filtered on best compound, best analogs, 85 best analog by using all this analysis. So just think that if you are really synthesizing this 10,000 compounds in a lab, it's really impossible to do this analysis. Impossible to do analysis. Okay, so if you want to find out a one specific substance from this 10,000 library of a compound, if you want to, by experimental lab, it's highly impossible. Just like finding a needle in a haystack. Okay, so this computational methods is highly helpful for us to narrow down the background noise. It is not specific for one particular discipline. Okay, this computational approaches can used anywhere. Even I'm just talking about the genetic terms. So every day we will receive so many samples, patient samples. We'll perform the whole exome sequencing, whole genome sequencing. When we do a whole genome sequencing, we will receive nearly 1 million variants we will receive. From the 1 million variants, we have to find out what is the specific causing variant. So if you want to do manually, it's highly impossible. Human DNA consists of nearly 3.2 billion base pairs of 3.2 billion base pair. Finding a one specific causing variant manually, it's highly impossible. You can't do manual step. We have to depend on these computational methods. These computational methods are the future. If computational methods are not there, to understanding the biological data, it's becoming very, very highly complex for us. So even I'm just talking about the basic research of what I had done. If the computational methods are not there, we can't do anything, any of the steps. Okay, so this is the thing I used. You can see here, you can use the molecular docking approach anywhere. So here we are going to perform the virtual docking also. We're going next demo session, we're going to do the virtual docking method, where you can add multiple ligands to a receptor molecule to find out the best ligand molecule out of the library of a compound. Okay, so these are some of the papers we published with the resveratrol molecules. We published in a Springer, and these are the old papers when I started my PhD days. At the time, these are some of the papers we done. Okay, so let us start this one. So the aim of the molecular docking is to give a predictions of the ligand receptor complex by computational method. So generally molecular docking can achieved by the two major integrated method steps. Two integrated steps. One is the sampling method, and second one is a scoring confirmation. What is sampling method means? Sampling method is algorithm that should be able to produce able to reproduce the experimental binding mode. So whatever the algorithm they develop, you can see here there are different algorithms are there. The algorithms are generally developed based upon the experimental evidence. Okay, so you already heard that last few days you have heard about artificial intelligence and also machine learning approaches. Okay, so all these apps or whatever the apps they are developing based on the already trained data set. They have been trained with the specific data set to get the accurate results. So once you have trained, okay, once you are trained, if your prediction structure, it comes in the same mode, then it will give a scoring function for you, specific character of a scoring function for you. There are two interrelated steps are there in docking. One is the sampling method, and the second one is a scoring function. Scoring function should be also rank its highest among the all generated confirmation. If you see here we have a various types of sampling algorithms are there. Okay, so nowadays so many docking tools, so many softwares are available. They have their own algorithm, they have their own methods are there. So first of all, you can see here matching algorithm. This matching algorithm are based on the molecular shapes. They use the space feature and chemical information of the ligand to map onto the active site of the protein. Okay, so here these are the some of the software. These are the some of the tools. Dog, Flog, LibDog, and SnapDog. They use the algorithm called as a matching algorithm. And the second one is the incremental construction algorithm is there. It is generally insert the ligand into the active site in a fragment and incremental fashion. Okay, it means so here it is going to form it will break down the ligand into several fragments and perform the docking. Okay, so out of them, the FlexX is one of the popular one. So we also generally use this tool. Okay, so here few more are there, and also Monte Carlo is there. Monte Carlo is one of the Socratic search algorithm, one of the basic algorithm. In previously Autodoc when started, they supposed to use this Monte Carlo searching algorithms. So now the Autodoc and Autodoc Vina, they are using this genetic algorithm, which is generally based on the Darwin theory of evaluation. The basic principle of the genetic algorithm is it will work based on the Darwin theory of evaluation, and this is an idea based on the language of natural genetic and biological evaluation. And finally, the molecular dynamics simulation tool, one of the best tool. And here, the best softwares are the Gromax and NEMT. So out of all the tools, there are so many tools are there. I just mentioned based upon the algorithm. I just considered few. Nearly more than 700 to 750 different type of online tool softwares are there. So here, what I considered is Autodoc Vina is a commercially it's academic free, and it is the most cited software. Even you can use the Gromax. Out of all the tool, I always prefer to go with the Gromax. But the problem with the Gromax is you need to have very high computational facilities, and you cannot work with the simple laptop or like desktop. You need to have a high-end servers to run this molecular dynamics simulation. Okay, so here the scoring function is generally in Autodoc. So Autodoc Vina or Autodoc, it determine the pairwise atomic energy. So whatever the value, whatever the score you're going to see, it is based upon the sum of it. It is a sum of van der Waal energy, hydrogen bond energy, and the dissolution energy. So once the results has been generated, I will show you how you can calculate this results part when you finish the Autodoc Vina results. Okay, so here, so once again, this Autodoc Vina, it is one of the best tool for the virtual docking. Okay, virtual screening with the Autodoc. With the Autodoc, you can do with one at a time. You can do what the single molecule. But in Autodoc Vina, you can do the multiple ligand you can use at the same time. So that's why we call it as a multiple virtual docking approaches. So at a time you can use based upon your system configuration. If you have a high-end server, you can run up to 50,000 compounds in a single shot, or 100,000 compounds in a single shot. So if you are using ordinary system or computers, my personal advice is don't go beyond the 500 ligand molecules. It will hang your system. You're not able to do anything. So here, virtual screening based on the molecular docking is one of the most powerful tool to identify the lead compounds in a huge pool of a small molecule. Okay, so as I said earlier, so scoring function used to calculate the free energy of the binding between protein and ligand molecule once the docking confirmation has been generated. Okay, let us start. So what exactly the prerequisites? What are the prerequisites of the protein ligand docking using Autodoc Vina? You need to have a Windows 10 or Windows 8. We have to install the Ubuntu terminal and MGL tools, Autodoc Vina, and Recepta we need to have, and link ligand molecule and open Babel tool. So don't worry if you don't have this the Windows 10 or Windows 8 in your system. So I will suggest you. I have my own channel is there. Uh, so where here I mentioned about the Autodoc Vina, but the older versions. Okay, so if you are using the Windows 7 or other versions, it is I think you can check my YouTube channel. So for other users, if you are the Mac users or if you are uh, Linux users or Ubuntu users, this is the way we can do this one with the these are the prerequisites that we needed. Okay, so here we need to have a receptor. We are going to retrieve the sum of the ligands, okay, and we're going to install all the tools like Open Babel1. And what are the file formats are there? So we have three different file formats are there. One is a PDB, protein data bank format file, and PDB QT, and that is a protein data bank partial charge atomic file, and one more structured data file of the drug molecule. So these are the three files we are going to get from this one. So we will consider one example, that is a human tyrosyl tRNA synthetase. It's one of the enzyme which is helpful in a translation mechanism, okay, which is helpful in a synthesis of the protein, okay. And it is already known that the compound resveratrol compound is going to bind to this protein and inhibit its action. So what we will do is, first of all, there are two options are there. One, for number one option is we can design our own drugs, or we can look for the substrate of our compound in a databases, okay, or analogs of this our compound in a databases. First of all, we will retrieve the analogs from the database, okay. Then we will retrieve the receptor from the database. Then we will start to perform the molecular docking. Okay, so let us start. So here I'm going to go to database, Jink database. Maybe might be you heard about this Jink database. Okay, this is a Jink database. Nearly it consists of a 21 million drugs are deposited here. So sorry, no more than 230 million drug drugs are available in this database. Okay, click here on substance. Okay, so if you click on this substance, it will display a menu for you. So here it's a Java based molecular panel, and also you can draw the structure here, and you can type your compound name. For example, if you type the resveratrol here, okay, just click on the resveratrol. Based upon the name, it will search and it will show you a number of compounds for you. Here we have 26 compounds are there. 26 compounds are there, but I can see I just want a few more, like I will take 100 compounds. So there is one more option. You can draw your structure here. For example, I'm drawing the structure here. Okay, so here the resveratrol compound structure here. So here click on substrate. Now you can see here nearly 99 compounds has been generated with this substrate. These are the analogs. These are the resveratrol analogs, which is almost the seed structure is same, but you can see the so many linkers are there. Okay, so now we have one in SDF format file. You can see SDF format file here. Okay, so here on desktop, you'll create a folder, docking. Okay, so here save this one. So now we save all the ligand molecules. I mean, all the 100 substrates or 19 substrates we save. Now next thing we have to download the receptor molecule. So here go to RCSB. Okay, so here I already pulled out the PDB ID. Okay, so here crystal structure of resveratrol bound human tyrosyl tRNA synthase. Okay, so here download this one PDB format file. We can save this one in the same folder. Okay, so here we save this file. Now we have two files. One is the ligand library and 100 compound of ligand library and the protein molecule, that is a receptor. First of all, we have to edit the receptor molecule. So directly you can do the edit here in the WordPad. Okay, so go to right click here open with WordPad. So come to the bottom of this page, and you have to look for the heteroatoms. Heteroatom, starting position of the heteroatoms. So here atom means this is a protein. So the problem with the major issue with the Autodoc or Autodoc Vena, when you're preparing a receptor molecule, it need to have only amino acids. Other than that, if you add any water molecule, if any water molecules are there, if any drug molecules are there in your protein structure, it's going to give a result. It's going to give a error for you. Okay, you always need to remember you have to remove the atom which is other than the amino acids. Okay, so here you can see here there is a term is it mean termination of the protein, and it is starting position of the heteroatom. I mean different type of molecules are there. Even the water molecules also we don't know. So remove until the end. Okay, so I removed them and I saved them here. Okay, click on save. Okay, so now we have a receptor ready and the substance ready. And next we will install the softwares. First of all, we have to install MGL tools. Type MGL tools in a Google, you will find the first link itself. Okay, click on download here. Now you can see different versions are there for different operating system. Whatever the operating system you are using, you can set up that one. Just you have to download this one. Okay, click on download option here, and it will automatically install. You just need to follow the same instruction as Windows will do. It will install the Autodoc for you. So once you install, just type Autodoc to be confirmed whether it installed properly or not. If you see this menu, this pop-out menu, it means your Autodoc program is installed properly. Okay, so next we will install few more tools which is helpful for us in a virtual docking method. So first of all, the Windows 10 or Windows 8 users, they have to go to this Microsoft Store. And here type Ubuntu. Type Ubuntu here. And the first link you have to select the first one. Okay, so click on install. It will take some time for you. It is almost 200 to 300 to 500 MB of space it is going to take, depending upon your internet speed. It's going to take some time for you. So once you've done this one, click on launch. Okay, so once you launch this one, you have to come to this terminal. This is called as a Ubuntu terminal. Okay, so this is the one why we are using this in the Ubuntu terminal. We can do these things in a Windows also. So you just need to remember whatever the software, whatever the software presently we have for the CAD, and also for even for the other bioinformatic data analysis, the Linux is a best platform. It is a most stable platform for the most of the softwares, most of the free software. If it is the commercial one, you will see the stable version in the Windows. But I always prefer to use the Linux based one, which is always giving a very stable results for you. Even whatever the tools are, I'm going to tell you, those tools are already available in a Windows, but they are not stable. You will find always some errors are there in that one. Okay, so let us check this one. So here, once you've done this one, so next step is we have to install some of the basic things in this terminal. So first we need to update this one. So we have to click sudo apt get update. So it will take some time for you. Once you are started initial phases, maybe it will take a few minutes for you to install all the updates. So next one, sudo apt get upgrade. Okay, so once you've done this one, automatically. Here I already installed the updates. That's why I didn't see any changes in the files. Okay, so now we updated the system. Now it's ready. So next step is we have to install the Open Babel tool. So this Open Babel tool is a very good tool for us. First of all, we have to convert our PDB molecules into PDB-QT. First of all, we have to minimize the ligand molecules. Okay, so first of all, we have to minimize the ligand molecule, and we have to convert them into charge molecule, the PDB-QT molecule. That is called as that is the molecule which is helpful for the docking mechanisms. So here we are going to install that one sudo apt install Open Babel. Okay, so I already installed that one. Open Babel already installed. If you're not installed this one, you will find within a few minutes you can to install the Open Babel. So next one is, now we installed the Open Babel. Okay, so next one is, if you want to cross check that one, just type OBABEL. Okay, if you type this one, you will find this menu. It means your system is properly installed. The Open Babel has been properly installed. Okay, so here let me clear. So next one is we have to install Autodoc Vena. Okay, sudo apt install Autodoc Vena. Okay, so you can see here I already installed the Vena. If you type Vena, so it will display this message. If it is displayed this message means so your system is properly installed. The Vena is properly installed. Okay, so now all the softwares we install. Along with this one more thing we needed. We need a PyMol. Okay, so this PyMol, you will find the academic free version here. If you click on the download, they will show you the academic free version. You have to provide your academic email ID. They will give the full license for you to use the academic purpose. Okay, this software is also must for us to molecular visualization. So once you install all the softwares, the terminal, MGL tools, and also Open Babel tool, Autodoc Vena, and the PyMol. So now you're completely ready. Now we go for the analysis process. Okay, so now we have here, you can see the receptor. When we calculate receptor, we have two different files are there. One is substance on receptor. This substance consists of a merged of 199 molecule. Now we have to extract all the 99 molecules. We have to extract all 99 molecule and save as a separate file. Okay, to run that one, we have to use the option called as a Open Babel one. Okay, OBABEL minus I SDF format file. So what is the SDF format file? We have substance. Sorry. Before that, we have to go to the path. Okay, we have to go to the path. How we can go to the particular path means you have to go to cd backslash. If you type the cd backslash, it will go to the root point. Okay, if you now click on the ls, so now you can see the different directories are there. Now click on the cd mnt mount point. If you click on the dir, it will show you different type of drives for you. CDE. Okay, so desktop will be appears in always C drive, C, and my user, and my desktop is located on user, and here desktop. Okay, now I'm in desktop. Okay, so I just want to locate to the CD docking library, docking folder, CD docking. Now you can see here I can find my two files. I can find my two files. Now we have to split, we have to divide, we have to extract 100 compounds in separate files. Okay, so to split that one, we have to use OBabel BABEL minus STF file. Input file we have to, what is the input file here? So input file is a substance STF, and output file, what type of output file we want. Generally here it will give you a SDF file for you, SDF format file, the output file we need, and capital O file name. What is the file name you want? We'll give the same file name, whatever the previous file name is there. Okay. And here, split. Click on split. Okay, as you can see here, we have 100 molecules has been converted. Let us see that one whether install. You can see here now all the 99 molecules, has been all the 90 molecules has been extracted from that one file. All the 99 molecules has been extracted from the one file. Okay, so all the 99 molecules has been extracted from the one file. So now we extracted this one. First of all, what we need to do is after extraction of all this compound in Autodoc Vina, we have to minimize we have to give the energy minimization for the both receptor and ligand molecule. Energy minimization is very important step to remove the bad contacts in a protein or ligand molecule. You always need to allow the some forces, add some forces to the ligand and receptor molecules is highly essential. Okay. So what we'll do is now we'll prepare a one more command. Okay. So here we'll write ob. This is also from the same open babel. Okay. Ob minimize. So ob minimize. Okay. If you type ob minimize, sorry, ob mic. Okay, if you type this ob minimize, it will show the input file and output file for you. Okay, here we have a 5 different type of force fields are there. Force fields are there for the ligand molecule. So out of the 4, we will generally use for the, if you're using for the docking purpose, we are going to use this MM494 force field. So don't worry about the force field. Force field, I'm not going to discuss about the mathematical terms. It's just force field, as I told you, it will restrain the ligand molecule. Whatever the bad context is there, it will remove the bad context in a ligand or protein molecule, and it will all the charges are distributed equally. All the charges will be distributed equally towards the complete atoms in a ligand molecule or in the receptor molecule. Okay, so here we'll type the ob minimize. So we are going to use the force field MMFF94 force field and minus n. How many runs we want? 1000 runs, and I'm going to minimize for all the SDF file. Okay, so here it is it completely minimizes the structures of all the structures of the ligand molecule. Now our ligand molecules are ready. Now all this SGS molecules has been charged. Next step is we have to convert this SDF format. Autodoc cannot take directly SDF format file. It has to convert as a charge format file that is called as a PDB QT format. PDB QT format. This is also a very important step here. So again, we have to type Babel. Okay, so Babel. So input file minus ISDF. Okay, star.sdf file. SDF file. What is the output file you want? Minus P. What is the minus O? What is the output file format you want? Okay, I want PDB QT. I want PDB QT format file. And what is the file name you want? This is a capital O. Capital O star dot PDB QT. PDB QT. You can see here all the 99 molecules has been converted. Okay, so all the 99 molecules has been converted. So if we click here, sort by type, it will display the PDB QT format file for you. Okay, so here you type the format. You can see here the PDB QT format file. So at the end, you will find all the SDF format file. Now we don't want this SDF format file. Okay, let us save this one in a separate folder. Okay, so here I'm cutting this one folder. Okay, so I'm going to create back. So here now we have the PDBQT format files, like in all the PDBQT formats are ready. So next step is we have to prepare the ligand. Sorry, we have to prepare the receptor molecules. Okay, so now ligand preparation is done for us. Next step is we have to prepare the receptor molecule. Okay, so for this you have to go to Autodoc. So click on remind later here. So go to file, read molecule. Okay, so here this is our receptor molecule. Okay, so click on this one. So this is our receptor molecule. Okay, so here first of all we have to check whether any missing atoms are there in this structure or not. This is also very important step. Most of the people generally they'll get a lot of errors while they are performing the docking. So why it will get mean in when the protein is crystallized, when the protein is always you will find some amino acids are missing during crystallization procedure. Okay, if the missing amino acids are there, it's a problem for you. You will get a lot of error for you when you are doing the docking analysis. So always my suggestion is you have to repair this missing atoms, and you have to add charges to it. And once you've done this one, your protein will be ready. And I heard few more errors. I have a lot of errors from other people or students. They are complaining that sometimes Autodoc cannot repair the proteins. Okay, if you have a huge amount of huge protein is there, it cannot repair that one. At that time what you need to do is we have two options are there. Once you can go to Swiss model server. Okay, you can paste your sequence here, your protein sequence, and click on the build model. So once you do that one, it will build the homology model for you by adding all the missing atoms. Okay, this one is also another one, the mod web. This is also the mod web is also is there, where these are the two very free tools are there, where you just need to paste your protein sequence. It will generate all the protein structure for you with filling the missing atoms, by filling the missing atoms. But here I will tell you the method how to do this one. Okay, click on edit here once you load your protein structure, and click here misc, check for missing atoms. Okay, you can see here there are so many missing atoms are there. So many atoms are got missed. Click on select all residues, dismiss, go to edit one more time, misc. Okay, repair missing atoms. So this will take some time, maybe depending upon the number of missing atoms are there in your protein structure. It will take to 10 to sometime 10 minutes, or even the 15 minutes. Okay, if you have a huge protein is there, then it will always take a huge amount of time for you to repair the missing atoms. So once we created these files, the protein file and the receptor file, now next time is we have to check for the configuration file. Okay, so here here also maybe depending upon your system configuration, I always advise if you are working with the molecular doc, with the Autodoc, or if you're working with the multi-dynamic simulation, or if you're using the high library of a compound uh, in a CAD discovery and QSR studies, I always advise you to use the best configuration. At least you need to have a 16 GB of RAM in your system, or a i7 processor. We definitely need it. Okay, so you can see here, once we done this one, next step is we have to save all these files. So click here, save as two sets. Okay, save as two sets, click on dismiss and unselect this one. Okay, so unselect this one. So unselect this one. So once you've done this one, the next step is now we have to add charges to this protein. Okay, so before adding charges, one more very important step we have to do is we have to edit the histidine. We have to edit the histidine amino acid. So if you see the structure of the histidine, it consists of it has a ability to proteination and deproteination ability. It has ability for deproteination and proteination. So we have to neutralize this one. Always it consists of a deproteination or proteination state. We have to neutralize this all the histidine amino acid present in a protein. Why? Mean the charge should be distributed equally throughout the protein. If you have a +1 charge on histidine, you will find an imbalance of charge will be there on a protein. That's why we have to neutralize the protein with the same charge. So now we have to create this one as a neutral 0. Okay, click on apply and dismiss. Okay, so check this one whether the charge has been distributed equally or not. So sorry, now it's time to add a polar hydrogen atoms. Okay, go to hydrogens here, add polar only, click OK. Now it added all the polar hydrogens. Next, go to again edit here. It's time to add charges to this protein. So here we have to add a Kalman charges. Okay, so we have to add a Kalman charges. Click on Kalman charges. You can see here total charge has been added 7.958. So once again, you have to check whether the charge has been distributed equally or not. Go to edit here, charge, check for total residue on total on residues. So some of the residues has not distributed equally. Again, you have to click this one, spread the charge deflect over all the atoms in the residue. Okay, click on dismiss. Again, go to edit, check one more time, charges, check total. Now, if you find this one, no residue with the non-integral charges found, mean your charge has been distributed equally in the protein. So equally in the protein. So now next step is we have to find the ligand binding site. Okay, so go to grid box here. First of all, we'll save this one. Macromolecule, grid, macromolecule, choose. Okay, select this one. Okay, so you have to save this one as a receptor dot pdb qt. Receptor dot pdb qt. Okay, so here I save this one as a receptor. Now the receptor is done and the ligand molecules are prepared. Both are there. Now it's time to prepare the grid box. Okay, where exactly docking you want and which particular area your docking conformation or your ligand need to interact. So there are two methods are there. One is known grid box preparation and blind docking preparation. If your grid box is already, I mean, if your active sites are already known, you have to take the XYZ coordinates of the protein, and you have to create the grid box based upon your active site. So how you can do that one? You can check my YouTube channel. There I mentioned that if you have a already known active site, how you can calculate, how you can create a grid box. So we have very limited time here as a 1.5 hour. We cannot cover whole thing in 1.5 hour session. Okay, so I will show you the blind docking. Blind docking mean it will cover everything for you. So here, go to grid box here. Okay, so grid box. Now you can see here, this is a grid box. This is a box region. This is a box region where your ligand should be should bind. If you create with the same space here, maybe we are giving very limited space. We are giving very limited space to this one. Okay, so maybe it's not enough. We have to expand the box if you are working with the blind docking. Blind docking mean it need to cover whole protein. Okay, so let us change the dimension. So you just need to drag down this one. Okay, or otherwise you just click on the left click. Okay, here it will display a number for you. So I'm going to use the XYZ coordinates as a 80. Okay, here also 80. And here also I'm going to use 80. Okay, so apply. Okay, and you can see here almost it covered everything. If you think that if it is not covered properly, if you want to move this box, you can change this grid center. Okay, you can change this, you can move this grid center. Just I will move this one. Okay, here. So almost the protein has been covered, except this region is there lightly. So here I will, okay. Okay, so almost the protein is covered, almost the box is created. Now it's time to create a grid box parameters here. So go to desktop and type here text for document. Okay, open text document here. Okay, so open text document. First of all, we have to give the receptor name. Type receptor is equals to receptor dot pdbqt. Okay, so next one is we have to give, you have to type exactly. If you made any single error, it's going to give a error for you. Okay, so what is the center axis you have selected? Center underscore x center underscore y center underscore z. So center S we selected 80 here. Center Y we selected 80 here, and Z also the same thing we have done. Okay, so here, what is the size of the box? Size of the grid box we selected size underscore x is equal to minus 15.254. Size y is equals to 21.322, and size z or z equals to minus 8.797. Okay, so next one is the number of nodes. Number of nodes unit. I mean number of energy confirmation. Number of times your ligand should dock. How many number of times your ligand should be docked at the pocket. Okay, so generally we will give a default for the time purpose. I'm going to give only a 10 compound. You can up to, you can give 100 compound, 100 orientation also. Okay, and energy range. Okay, so what are the energy range you are going to use? We are going to add the 4. So once you do this one, you have to save this file as a. Okay, so once you've done this one, yeah, we have to save. We have to be very careful. You just want to cross check whether the name is correct or not. Receptor.pdbqt. So here I save this one as a RECEPTOR receptor.pdbqt. Okay, so now it's fine. So save this one as the conf conf, conf. Okay, so I'm saved this one as a conf. So configuration file I saved. Okay, so here the next step is we have to save all the file names. We have to save all the file names in a one text document. Okay, so I'll tell you why we need to save that one. So when we are writing the Perl program, generally Autodoc Vina, it doesn't have the automated things for you. It doesn't have automated to perform the virtual screening. Okay, so we have to write some script to it, to some script program to it to run all the ligand at a single shot. Okay, so I have written one script on a Perl. I will show you here. I will share you that Perl script with you. Okay, so here okay, this is a Perl script which I have written for this virtual screening. You can use this one for your studies also. It's not a problem for me. Okay, you can use this information, and you do not to change anything. You just follow the same thing. Whatever I'm doing here, you can run up to the 50,000 compound, more than 50,000 compound with the same script. Okay, so here the next thing is we have to convert this one. We have to convert them in all the file names. We want all the file names of the ligands. Okay, we want all the file names of the ligand in a text format. Okay, so we need to have a ligand in a text format. So we have a small command for that one also. I'm going to write a small command for that one. Sorry here. So everything I'm using this Ubuntu version only. If you, as I told you, if you are not familiar or if you are not able to install this Ubuntu terminal in your system, anyway, you can go back and check my channel where I clearly mentioned for the Windows 7 users and other users how we can generate all these files by using this lower versions of the Windows. Okay, so here the next command we're going to write is ls. Okay, ls. Okay, so here ls and write down as a ligand.txt. Okay, so let us check that one. Go to docking here. You can see here ligand.txt. Now it saved everything. All the text file name has been saved, but you can see here some of the things we don't want. We want only the ligand molecule names. Okay, so we want to remove these other things which is not required for us. Okay, we want only the file names of the ligand molecules. So save this one. Now we can save this one. The next option is the next command we have to write is about the Perl. Okay, let us start a Perl script. Okay, so now we have everything. We have configuration file is there. We have configuration file is there. Now we have ligand names are there. Receptor is there. Okay, so receptors are there. And like you can see here, 5 different files are there. We have one is the ligand name, and second one is confirmation file, and third one is the Perl script file, and receptor file, and all the other ligand molecules. Okay, all the files are there. Now it's time to run the Autodoc Vina. Okay, let's run the command. Very simple command. We have to write like Perl. Okay, so Perl is automatically installed in Windows. You need not to worry. If you want to check the version, click on Perl -v. will display a option for you. Okay, so here now we have to write the command here, Perl Vina Perl Vina underscore PL. Okay, hit enter. So what is the ligand file name we have given? You can see here the ligand file name we given as a ligand text. Ligand txt. There something is there. Configuration file press error unrecognized option number of nodes. So there is some error is there. We have to check this one. Number of nodes. So that's the thing. If you made a single mistake, number of not nodes, you can see here. So we have to type number of modes. Okay, even if you made a single mistake, it's going to give a result for you. So let's run one more time. Ligand.txt. Okay, so here I think while writing the command, while writing the text format file, I made that's why I told you. So whenever you're writing the text format file, the configuration file, you need to be very aware about it. Now you can see here all the programs are running properly now. Each and every molecule has been Vina docking has been carried out. Again, it's going to take a lot of time for you. I here entered nearly 100 compounds. I nearly entered the 100 compounds. Maybe it's going to take, for me, I have a 32 GB of RAM of some system. It will take me around maybe around 40 minutes or 45 minutes. It's going to take to complete the whole uh, docking, whole virtual docking. Okay, so I'm going to terminate this process. Why means already I've done this one. I already finished that all the 100 molecules. I will show you the results for you. Okay, so here I'm terminating this process. So here I terminated this process. Okay, so I will show you that for folder name here. So once you've done this one, once you complete this one, it's going to generate two different files for you. Okay, so here two different files it will generate for you. One is, sorry. Okay, so here two different files it will generate for you. One is a PDB log file and also the ligand binding site file. Okay, so you can see here, if you open this log file, it will open you, it will tell you what is the affinity, how much strongly your ligand has been interacted. So as I said to you, we have given 10 confirmation to it, 10 different confirmation to it. That's why you get 10 different forms of binding mode with the resveratrol molecule. Sorry, uh, with the NFKB, with the receptor molecule. Hello. Okay, so with the receptor molecule. So now we got this one. You can see this one. I will show you this one uh, in PyMol also. You just need to click this one. Okay, so here, file, open. So here we loaded the ligand molecule. Okay, now load the receptor molecule in the same folder, the same receptor molecule you have to give. Okay, so here, okay, so click here. You can see here, if you click this one, it will show you 10 different conformation for you. So 10 different conformation where your ligand has been interacting. So let us change this one. So here, preset ball and stick. Okay, so ball and stick here. You just need to click find polar context to any atom. Okay, so click on find polar context to any atom. If you have any hydrogen bond interactions are there, it will show. You can see here this conformation, it is binding, it is forming a 3 hydrogen bonds. This compound is forming a 3 hydrogen bonds with the resveratrol molecule. So here, go to this receptor and click on show lines. So where it will display the binding sites for you. Okay, so you have to highlight them. Okay, click on show sticks. Again, hide lines. You can see here clearly this ligand molecules is forming a strong hydrogen bond interaction with the receptor molecule. This receptor molecule, there are 2 hydrogen bonds are there. So the issue with this PyMol is it's going to show you only the hydrogen bond. If you are working on, if you are thinking the van der Waal interaction, other interactions are there, stacking interaction, any other weak interaction, salt bridges, you cannot find this one in the PyMol. You can see only the strong hydrogen bond interaction, which is nearly 2.5 angstrom away from your drug site. Okay, so here you can load multiple molecules. You can load multiple molecules at a time. So here, go to file, open. Okay, so here item type. So here I'm going to only out file. You have to load the out file, not the PDBQT file. Okay, so here, so you can see here. So I added the multiple molecules. You can perform. So you can see here. So all the 10 different conformations, 10 various conformation it is showing based upon the binding energy, based upon how much binding energy you got. Based on that only you can separate the molecules. Okay, so here as I told you here one more time, this is a step, this is the energy minimization step here. Sorry, this is a output file you're going to generate. This is a log file you're going to generate. So this is the log file, so where it is showing the binding energies for you, binding energies is for you. So here out of the all the 10 conformation, we have minus 7.9 for this 99 compound. So similarly, you can see this 98 compound. 98 compound also showing almost identical results as minus 78. So here minus 9 compound, let us see, it is showing the less binding affinity. So if it is more binding affinity, mean your compound having a very high interaction towards the ligand, towards the protein molecule. Okay, so here you can get comprehensive. If you want to have a comprehensive, you want to see all the results in a one text format file, you can get that one also. So you have to write a small command here. Small command here. So here, go to the same Ubuntu script. So here, just go to Ubuntu script, same one. Okay, so here we have to type, let's clear this one. Okay, clear. So now type this command, the small command here, tail -n 11 line star.log file, and we have to read this one into as a result, final result.txt. Okay, so here enter. So now we can check this one. Sorry, so file is here. Hit enter. Now let us check the same folder. So you can see results here. Now you can see it is grabbed all the PDBQT format files. So all the 199 PDB format file is grabbed, and it is showed very simple, the straightforward energies for you. Always you have to select the first one. First one is the best confirmation, first docking confirmation. So here you can see manually, we can see here the second one is going the good energy for us. And even here the 28 compound also shown the -8.4 kilocalories energy. And here -66 compound is showing the best energy compared to other compound. Like this you can filter down. It's here also you can use the Excel sheet. You can open this one in Excel sheet where you can filter down based upon the number of based upon the interaction energy, based upon the affinity of the binding affinity score. We can change, we can select the specific compound. Out of the 100 compound, you can see this 66 compound is showing the best docking energy compared with the other compounds. Like this we can narrow down. We can narrow down if you have a thousands of compound, we can easily narrow down to very few compound which is showing a best affinity towards the ligand molecules. Okay, so here this is about Autodoc Vena. Very straightforward tool, but a very powerful tool when compared with the Autodoc. So Autodoc, as I told you, Autodoc you can do only one compound at a time, and also there is a error ratio is very high in Autodoc. Why means you have to be very careful during the energy minimization, and also the distribution of charges in both the ligand and receptor molecule. And once, and also it's going to take us so much of time. It's not as quick as Vena. So if you want to run one component itself, it's going to take almost 20 to 30 minutes. If you want to run 100 confirmation, it's going to take 20 to 100 minutes, sorry, 20 to 30 minutes for us to complete the docking procedure. Okay, so here this is about Autodoc Vena, and I just if you have anything, you can just raise the questions to me about this topic.
Sir, chat box, please look, sir.
Sir,
One, please. I think organizer can ask. Hello.
yes, sir. So the very first question is from Mr. Sujit Das.
Yes.
If a new phytoconstituent is isolated and its protein is not known, how can we go forward with this technique?
Okay. So there is the one option. If the target is not known, if what he's saying is maybe target is not known, the drug has been discovered. Generally,
Yes,
drug
sir.
discovery generally it will take. If you have a target, then only you can discover the drug. Maybe Mr. Sujit is talking in reverse manner. If the drug is known, it cannot be validated by the target. If target is known, then only we are able to design a drugs against that one. You can, if you have any doubt, you can continue the same thing with Dr. or Mr. Sujit.
Next question is from Sindhu. On what basis analogs of resveratrol is generated?
Yeah. So on what basis the resveratrol analogs has been generated? So here, that's the thing I told you. If the natural compound, the resveratrol is not giving it's losing its bioavailability. So we want to increase the bioavailability of the compound. We want to increase the activity of the compound. On that base, we added a few more linker to increase the activity of the compound. Okay, that's why we added different type of chemical moieties to the stable compound. Resveratrol itself is a very stable compound. It's a natural compound. If you are able to add a linker, if you are able to modify the natural compound, still it will remain stable than compared with the organic, completely organic synthesis compound. So on that base only, yeah.
If
Yes,
we
please.
select the analogs of resveratrol for anti-cancer activity, what will be its effect on lipid levels of the body?
Not only lipid levels, it will, I think we can give a prediction as a bioinformatician. We can give a predictions to the society. Our study. Okay, like this. This is going to happen if you have consumed this compound. It's going to give a side effects for you. This going to give a this thing for you. We are going to do that one by computational. We don't know biologically, unless until it is released into the market, or unless until people start to take the drug, unless until it's having clinical trial has been done, unless until it's in vivo studies, we cannot predict that one. That's why in vivo studies is a very important one. Once your compound has been done, passed through all these filters, like in vitro studies and this computational studies, next step is in vivo studies. So pupils are generally, they use for example in cancer, they will take the nude mouse or a cancer affected mouse. They will try to treat with this compound. They will check all the profile, whatever the profile you said, but the lipid profile is there, how about the metabolic profile, everything they will check, and they will come to conclusion. So you cannot predict them. We are not sure that unless until you have studied in biologically.
Next question is from Mr. Keshav. Commands in Linux seem to be difficult to follow. Can we have this in Windows?
Yes, of course. Of course you have. I told you here, you can go to my YouTube channel. I provided all the links for you. But again, I'm suggesting you here. I did. I think I made a lot of effort on this one to prepare the pipeline. You don't get this same pipeline anywhere. Okay, so if you are able to use this same pipeline, it's a most stable system. You're going to get a very quick results compared to the Windows. You can try in a Windows also. As I told you, you can go and check my YouTube channel. We have a beautiful protocol on Windows also.
Next question is from Ms. Tanmay. Using Autodoc Veena, can we obtain glide score and docking score?
Yes, yes. We are getting the binding affinity score. What is other thing you said? Glide score.
And
glide
docking score.
Yeah, we will get the affinity score. That's the thing I showed you here. That's the binding energy. This is a binding affinity score. Okay, it's a docking score. Whatever name you call this, affinity score or docking score. This is based on this only. You can separate them all. You can calculate which particular ligand has high affinity towards the receptor molecules. Yeah.
So, which is a better software, PyMol versus BioVia Discovery Studio?
I never use this BioVia Discovery Studio for molecular visualization. I always prefer to use the PyMol. So I mean, it's easy to use and more easy to have a graphical interface. Very good graphical interface are there, and most of my publication always go. If I work with the protein, always I prefer to use the PyMol.
Is it possible to develop libraries or check docking of multiple compounds at once using Chimera?
Chimera using Chimera? Can you repeat the question, please?
So the compound analysis that you showed now, sir, at once you have checked 99 compounds.
Yes.
So is it possible using Chimera?
Yes. In Chimera also, generally, previously I did the energy minimization with the Chimera only. You can upload. You can upload, but you cannot split the molecule. How we did with the OpenBabel, like the one SDF file, you cannot split into multiple SDF format file, but you can do the energy minimization in Chimera.
Sir, can we do docking using Pyrex software?
Yes, we can. But I think still, I feel the Pyrex is always having some issues in a Windows. I don't know about their paid version. Now they have the 0.8 version. I think it seems to be. It's a free for everyone. If you load more than 10 or 15 compounds, it's going to struck or it's not going to give a results for you. It's okay. You can use the Pyrex, but I feel if I add more than 10 or 20 compounds for the virtual docking, I'm getting error. So it's not processing.
Chimera versus MGL tools, which would be better?
That is two different one. That's a two entirely different one. Chimera is for a visualization. We can do so many things in Chimera. Even you can add a different type of plugins to it, like a docking plugin. Even we can do the homology modeling also in a Chimera. All the molecular visualization, beautiful pictures, graphs you can generate in Chimera. But MGL tool is only specifically for the molecular docking. Nowhere can use for molecular visualization. It's a very odd. You can see the MGL tool visualization pattern. It's very odd. You can use for the molecular docking only.
Can you suggest best tutorials or MD Dynamics?
Molecular dynamics simulation.
Yes.
Yeah, of course. For molecular dynamics simulation, that guy name. For the GROMAX, I know I work with the GROMAX. I think I will give a link for you. I will send a link for the coordinators where you can get that information from them. Or you can just type in a YouTube discussion bot. I can answer you. So or otherwise you can, I don't want to promote myself. I have one tutorial on my YouTube channel. You can go there and check my tutorial on a GROMAX MD simulation.
Sir, how to proceed for reverse docking?
Reverse docking, I didn't have experience on the reverse docking. I don't know. I never worked with the reverse docking. Maybe they are talking about the vaccinology things. No, I didn't work with that one. Sorry.
Which is better, Schrodinger versus Autodoc Vina?
Schrodinger is a commercial version. You cannot compare with Autodoc Vina. And the annual license is nearly $10,000 for that one. You cannot compare that one. Always Schrodinger I feel it's the best one. But I think you cannot afford, the normal people cannot afford $10,000. Then they always have to go for the Autodoc Vina. If, as I told you, if you go for the publication citation, always believe on that one. If I am using any software for my study, I always check whether how many citations have that for that particular software. If we have a more citation or if it is published in a very good journal like in Nature or Science, I will go with that one. Okay. So we Autodoc Vina having so many citations are there.
Can we proceed with Autodoc even if it is having any metal or ATP in the binding pocket if the ligand is having?
You can proceed, but there is a limit is there. I think 23 atom they listed Autodoc 1. If the 23 atom list, if your metal ion is listed in the 23 atom, you can proceed with that one. If it is not listed, for example, as you said, the magnesium, it's not listed in that list. It means you cannot do that one. Some of the like calcium is there in that list. You can proceed with the calcium. Okay, there is some restrictions are there. You have to create your own carbon map. You have to create your own atom map. That is highly complicated. You have to have a Python experience on that one. You have to create your own script for that tool for the specific metal and atoms.
How many parameters can we study using polymer and anti-cancer hydrophobic drug using Autodoc Vina?
How many parameters can we study? How many is we only binding efficacy we can check what we can do. We cannot do anything. Only two things we can do. If you are working with the Autodoc, they are going to give extra parameters for you like inhibition constant Ki and RMSD values also they will give, and binding affinity score. These three values, and also hydrophobic interactions and van der Waals scores. All this information it will give by the Autodoc. If you are working with the Autodoc Vina, it will show you only two energies for you. Only is the affinity energy, and second one is the RMST.
We want to develop a coarse grain model of a receptor for our study. Could you please suggest any MD simulations which can be used?
Yeah, the Gromax ultimately the best one, and I feel, but you have to have a very high computational facility to perform the Gromax MD stimulation. And there are some Schrodinger, as this earlier person suggested, there is Schrodinger version also is there, but that's a paid version, and also it will support only for the Linux. The free version is there for the from the Schrodinger, but it will support only the Linux, not the Windows.
What is the best software for protein-protein docking?
Yeah, for protein docking, there are three softwares. There is no, I don't feel it's, I think, the best software. Still, I think the protein things is still under developing stage. But I can suggest three tools for you. One is a ClusPro, and PatchDock, and Hex docking server. So these three are best one. I personally use for my studies these three tools. So these three tools has been well recognized. You can use them for your docking purpose, protein docking interaction studies.
How to analyze the results obtained from docking studies, for example, energetics?
Yeah, so that's the thing. First, we have to find out what is the binding affinity is there. As I told you here, so you have to find the lowest binding energy out of all the compound. You have to find out what is the lowest binding energy. Second thing, you have to find out where exactly your ligand has been interacted, whether it is interacted in the active site of your protein, non-active site of your protein, and how many how many hydrogen bonds, how much strongly your ligand has been interacted with the protein. What are the bonds are there? How many van der Waals interactions are there? How many hydrogen bond interactions are there? How many weak interactions are there? So all these things you can analyze that one.
Which software is better to study polymeric drug delivery analysis?
Polymeric drug delivery. I mean, what study they want? I don't know. The question is wrong.
Sir, as you said, we can use Schrodinger using Linux platform. Is it the same way that you used right now using Ubuntu?
No, it is not straightforward. No. It's GUI based, graphical user interface based. You have to install. First installation itself is too complicated. You don't find any tutorial anywhere about that Schrodinger free tool. I just remember that one. Let me check this one. Schrodinger.
Sir, could you please make a video on that in your next?
Actually, I don't. Yeah, I don't have a time. When the COVID is happened, at the time I release a lot of videos. Now people are requesting me to prepare the videos. Now I'm busy with my own work and also with the students here. So sure, soon. I think soon I'm going to working on it. Maybe soon.
Do you provide any research internships or any kind for any online training or research internships for the students, sir?
Research internships in Saudi Arabia, we don't have that one. Maybe it's there in so many like US, they are providing so many internships. Yes, I will provide the links for you. I have few. I can provide the links for you. But regarding the tutorial means nowadays, I think many tools are available. Like you can go to Udemy if you are. My personal suggestion for the student is always learn this bioinformatics. You can apply anywhere. As I'm giving my example of mine, I actually trained in the CAD and also computer drug designing and also protein modeling and docking, dynamic stimulation. So, but basically I have a slightly programming interest. Then when I move to this Saudi university, here I have to adapt to the genetics environment. I'm working in a genetics lab where I need to redesign myself towards the genetics. So my suggestion to students is learn some basics in the programming. You can apply anywhere. So the demand for the bioinformatics is becoming very higher day by day. Why means this is not the basic field. This is application oriented. You can apply this field anywhere. If you have these skills, you can do anything. The future is belongs to the bioinformatics. The future is belong to artificial intelligence. Yeah. So regarding that tool, the Schrodinger tool, someone asked, that is a Desmond. That's a tool name called a Desmond. In future, I think I will prepare a video on that one, how to install that one and perform the indiscrimination in future. Surely.
Thank you, sir. That's all from the chat box.
Okay.
So the Autodoc that you are used is whether it is a free version or the purchased version, sir?
No, it's a free man, free. So academic, everything is free. Whatever the tools are discussed here, it's free.
Okay. All are free.
Yeah, everything is free.
Okay. Thank you.
So anything, any doubts, further doubts? If we have any further doubts, you can raise to me. You can send an email to me here, or you can, I'll just type my email here. So you can note down my email if you have any query regarding the bioinformatics. So whatever the field it is, whether it's a CAD or either it's a genetics or whether it's related to some other topic, you can just post an email to me. Mostly I can respond immediately to you.
Sir, any software from Schrodinger is free for academic purposes.
That's the thing. Desmond is free. And that the Schrodinger drug discovery studio, I think that's also from Schrodinger. That is also free, I think. It's for visualization.
Thank you, sir.
Yeah. Okay. So here, this is my university. I just want to show you some pictures of my university to you. I'm staying here since the last 7 years. I completed my PhD in SK University, Sri Krishna Deva University, Anantapur, and I came here since last 6 years. I'm working in this center. Here we have a huge facilities. The problem here in Saudi Arabia is even though we have a huge facilities, even you don't find this kind of facilities in a big institute like Harvard and Cambridge. We have a lack of human resource. Lack of the people who's expertise in the field. So fortunately for me, I got an opportunity to work here. We have a very great team. This is our wet lab. One of my wet lab is there. And this is my team. I would like to thank my team member. I am here. And this is my team member. This is my professor who is the head of our department. And I would like to thank our external collaborators who is from Queen's Mary University of London, the biggest personality, Professor Panos Telukas, and also the Majid Lako from Newcastle University. And thank you once again. Thank you so much for your attention, and thank you for organizers for inviting me here. It was a pleasure and honor to me to be with you.
Thank you, sir. Thank you, Babajan, sir. Thank you for your gracious presence and excellent explanation in this session, sir. It is a very informative session, sir. Sir.
Thank you. Thanks. Yes. Yes, ma'am.
Sir, so I am giving some conclusions about your session, sir. Sir,
Sure,
giving great insights in molecular docking. We appreciate your patience and detailed explanation about different computational methods. Sir explained about resveratrol analog synthesis and about 30 to 35 compounds, sir, how they are analyzed by using these computational tools and computational methods for understanding biological data, docking mechanism related to sampling methods and scoring schemes, and the importance of Autodoc Vena, which is used as a multiple virtual docking approaches. And we are also learned some things some these tools about the Autodoc Vena is a Jing 15 and Linux based tools. And I think all the participants learned how to operate Autodoc tools like Ubuntu and different another tool is ModeWeb. So how to write commands is very important in this thing.
Yes.
I request
Yes.
all the participants to go to YouTube channel and also Sir YouTube channel and participate the Autodoc Vina, practice the Autodoc Vina tools and practice ligand preparation and receptor preparation and docking methods. So I request all the participants join valedictory session and afternoon session. So thank you, sir. Thank you very much.
Thank you, Dr. Swadika. Thank you so much.
Thank you. Thank you, sir.
Thank
Thank
you,
you. This is the only one session where we received the more and more question answers, sir. So
Oh,
I'm very much delighted as the participants are very, very active and they are interacted very much with you. Okay. For that, I
thank you
am
so
very much happy, sir. And
thank you so
apart
much.
from
Actually,
that, sir.
sorry, ma'am. Actually, I thought I just want to give some basic talk to it. I don't know whether we have to add some extra, like more advanced course. Suddenly,
Yes.
I thought, okay, let us give some basic course to them. When next time, Mr. Varalakshmi suggested that there are some doctors also involving in this
Yes,
this
sir.
conference. So next time, I think we will discuss on the more advanced features.
Thank
Whatever the
you,
things I
sir.
am working here.
Sir, right
Next time
decision you have taken, sir. This is the right topic that you have taken, and you have demonstrated very well and explained crystal clearly how the molecular docking will be done by using the auto docking system. For that, we are very much thankful, sir. So we thank our student Varalakshmi, especially because she is the one of the bright student of our institute, and she has given you a reference, and I think our participants are also 100% satisfied with your demonstration also. Thank you, Varalakshmi. Thank you very much. So this is the right platform to convey my thanks to you also. Okay. So thank you, sir. Thank you very much. And a small request, sir. Is there any possibility for the collaboration at the MOU with your institute, sir?
Let us discuss privately, ma'am, instead of here.
Privately, sir. That's why.
Yeah.
We will discuss and we will mail you through our authorities also. Thank you, sir. Thank you very much.
You're welcome, ma'am. Thank you so much.
Thank you. And dear participants, afternoon at 3 o'clock we are having test, okay, and the link will be open like yesterday evening, that is 3 o'clock, today's evening 3 o'clock, and it will be closed by 3:30, okay, and this test is followed by a valedictory session, and it is just for half an hour. Be patient, and by that way we will complete our sessions. And other announcement is, Amma, Pooja, you just ask those names whether they are responded or not.
Not it, ma'am. I just got.
You just announce those phone names. Otherwise,
Gino, ma'am. Jino.
Gino. Okay, next.
DCSC 7C85, ma'am.
Who is that? DCSC 7085. Who is that? Otherwise, you will be your name will be under absentees. Will you post in the chat box or in the WhatsApp?
They can actually reply in the WhatsApp group, ma'am. I have enabled the option so that they can.
Enabled?
Yes, ma'am.
Okay, you just say that up to 3 o'clock we will wait for that. After that we will upload the attendance, and all the names, all the phone names will be the absentees only if you will not respond. Okay.
Okay.
This is for your information. Thank you. We will meet sharp 3:30. Okay. And 3 o'clock, all should be alert.
Excuse me, madam.
Who is this?
Madam, myself Keshav from Nagpur.
Okay, tell me, sir.
Actually, sometimes I have network issue at my end, so it may take couple of minutes extra to submit the Google form of exam. So please allow that.
Okay, okay, sir.
Thank you.
Okay, sir. Anything else? Any doubts? Okay. Okay, thank you once again.